to welcome Honorable Pro-Chancellor Lieutenant General V.K. Sharma, recipient of Ati Vishisht Seva Medal, Honorable Vice-Chancellor Professor Dr. R.S. Tomar, Honorable Registrar Dr. Rajesh Jain, today's chief guest Dr. Manish Kumar Chaurasia, Senior Principal Scientist, CDRI Lucknow, keynote speakers, our Director, Professor Dr. Manoj Kumar Mishra, Deputy Director, Professor Dr. Mukesh Singh Sikarwar, Organizing Secretary, Professor Dr. Neeraj Mishra, respected dignitaries, deans, directors, head of institutes, head of departments, faculty members, staff members, our valued delegates, and dear students who have joined online as well. Today, I, Roshni,
And I, Arpit, extend a hearty welcome to the National Conference on Artificial Intelligence in Drug Delivery and Design, from innovation to implementation, organized by Amity Institute of Pharmacy, Amity University, Madhya Pradesh.
Today, we stand at the threshold of a transformative era in pharmaceutical science, where the convergence of artificial intelligence and drug development promises to revolutionize how we approach healthcare. We are here to explore, discuss, and deliberate on the cutting edge advancements in AI and its profound impact on drug delivery and design.
This national conference is the platform where brilliant minds from academia and research institutions converge to share insights, breakthroughs, and best practices which contributes from experts across the field. This event aims to accelerate the translation of innovative ideas into tangible solutions that benefit patients worldwide. I request Honorable Pro-Chancellor Sir to welcome Chief Guest Dr. Manish Kumar Chaurasia, Senior Principal Scientist, CDRI, Lucknow. Sir, please. Thank you, sir.
I request Honorable Vice Chancellor Sir to welcome keynote speaker Dr. Sanyog Jain, Professor and Head of Pharmaceutics at NIPER Mohali. Sir, please.
I would like to invite Director, Amity Institute of Pharmacy to welcome Honorable Pro-Chancellor Sir. Sir, please.
Thank you, sir. I'd now request Deputy Director, Amity Institute of Pharmacy, to welcome our Honorable Vice Chancellor. Sir, please. Moving ahead towards the conference, let's take the blessings from Goddess Saraswati. To invoke the blessings of Goddess Saraswati, the goddess of wisdom and learning, let us light the lamp of knowledge. For this, I invite our esteemed guests, organizing secretary, and dignitaries to light the ceremonial lamp.
Thank you, esteemed dignitaries. We would now like to invite our Honorable Vice Chancellor, Professor Dr. R.S. Tomar, for his welcome address. Sir, please.
Honorable Chief Guest of today's inaugural session of National Conference on Artificial Intelligence in Drug Delivery and Design from Innovation to Implementation. There is Dr. Chaurasia, who is the Principal Scientist in CDRI Lucknow. Keynote speaker, Dr. Jain from NIPR Mohali. Pro-Chancellor, Lieutenant V.K. Sharma, Aviation retired. HOI, Dr. Manoj, and Deputy Director, Dr. Mukesh, the Deans, Directors, HOIs, faculty members, and my dear students. This is the era of AI/ML. You all know what is AI/ML, artificial intelligence and machine learning. Things are changing very fast due to interference of AI in almost all the fields of day-to-day life. So is the drug delivery and design. I feel this particular topic that is chosen for the conference quite apt in today's context. The time is changing very fast, and we have to cope up with that. We have to make a pace with the change in the time. So AI/ML is the future of, I should say, not all, but most of the things. We are truly honored to host this landmark gathering of scientists, researchers, healthcare professionals, industry leaders, and students who are all united by a common cause, that is to discuss the role of artificial intelligence in drug design and delivery. You all know what is drug. Any chemical that is taken in a prescribed dose, which can bring about some desirable biological change, is the drug. The drug become harmful also sometimes, because if you take the more than prescribed dose, then it can be toxic also. So drug design and delivery itself is a big challenge. Today, when we are talking about gene therapy, gene editing through CRISPR technology and all that, I think designing the drug and its delivery is a big challenge. Because whenever any chemical goes inside our body, it has some or other indirect effect or direct effect on the body. It is healing one organ, but affecting many others simultaneously. That's why we are talking about the holistic treatment, by which we can minimize the effect of chemicals on the human body. You all know the drug design process is a very complicated process that comprises 4 primary stages, in which the most important is the basic research. The foundation lies with the basic research, And then we proceed to the next stage, that is the preclinical trials, then clinical phase, and post-surveillance, after the drug has been used for some treatments. So after the successful completion of these stages, the drug is finally approved for production. This comprehensive process typically spans approximately 10 to 12 years of period. So AI is required. Because to treat any disease, if you require 10 to 12 years to develop a drug, I think by that time, that drug will also become obsolete. The way the microorganisms are changing their body, their physiological activities, we have recently seen in the COVID-19 time. That time, not much AI was used because AI actually, the use of AI in drug design delivery actually started only after that. But we expedited the process of development of vaccine by doing it in vitro method. In vitro method is well-established method by which instead of directly targeting the animal, you are taking about any part or any body fluid of the animal, and then you can design a vaccine. So that was the major role played by the Serum Institute of India, you all know. SII, that is basically known for the storage and collection of the serum. So through that, the vaccine was developed fast, and anyhow, India was in the leading countries that counter the effect of the COVID-19. If this would have been the application of AI also, I'm sure it could have been more faster. So this is the role of AI. Now coming to the specific applications of AI in drug design, many things we discussed today through various experts and scientists. In this, the major applications are antibody design, small molecule activity prediction, new antibiotic compound identification, and drug repurposing. These are the basic needs and requirement of today's time. So the role of AI in drug delivery is also changing. That is, the future of AI-driven drug delivery lies in its integration with other emerging technologies like CRISPR for gene editing, blockchain for secure data management, and the Internet of Things for real-time patient monitoring. So altogether, they will play a role in the drug delivery. So AI/ML have great potential in drug development. So now the boundaries of the disciplines are breaking. You cannot stick to the particular discipline that I am from pharmacy or biotechnology or from this field or that field of sciences or technology. I need not to be dependent on the engineering aspects. Similarly, the engineering people, we have to be dependent on the other sciences also. So that way we can actually work together in a synergy and can bring about some drugs which are really effective and less time consuming. So need of the hour is this, that how we can develop the drug in a faster manner. So in short, I should say that AI is transforming drug delivery by enabling more precise, personalized, and efficient therapeutic interventions. They are leading to better patient outcomes and driving innovation in the pharmaceutical industry. But there are some challenges also at the same time, like one of the key challenges is availability of suitable data. So data is the future oil, you all know. So where we shall get the data, all will depend upon that. Then second challenge is the ethical considerations. Ethical consideration, when data is shared, then everything is shared. So we must be careful that if any data of any patient is shared, the ethical considerations are also a big challenge for the scientists and the researchers. But I must tell you in short that future is AI. I give you very latest example that I read day before yesterday on social media, in which one boy, 3 years old only, named Alex, he was suffering from some toothache. continuously from 2 years of his lifespan. 3 years of life, from last 2 years, he was suffering from toothache. The mother visited 70 doctors, dentists or this and that, oropharyngist, and there are many specializations in the dentistry also. So she visited 17 doctors, but none of them actually could treat the child permanently so that he can actually get the relief, actual relief. Somebody were giving the analgesics or some other kinds of medicines. They were giving him temporary relief, but not the permanent. So what mother did? She actually put up all the symptoms on the Google with the help of AI, and she tried to search that what exactly is affecting my child's dentition. And surprisingly, she found that he is not suffering from any local problem. He is suffering from some neurological disorder. And that neurological disorder, when she actually pinpointed, then she consulted a neurologist, not to a dentist. And there she explained all these things because she was raw in this field. She was not sure. But yes, doctors can easily confirm this thing. So doctors further did the other investigations in which you all know, MRI. Through the MRI, doctor could relate that particular problem with the neurological disorder, and then the treatment of that child started with the neurological problem, not that of toothache. And within 1 month, the child was completely cured. So this is the latest and good example of the AI use in the treatment process. So the future of doctors is also a big question. If they will keep on doing like this, the patients are going from pillar to post, not getting treated, then people like us who are not doctors, they can search on Google, putting the symptoms, and they can actually assess the doctors. Doctor Saab, I think I should go to a particular doctor, not to you. So this is the future of AI/ML. Very soon you will be all observing it. The young generation, you will face a lot many challenges because AI is interfering or involving itself in each and every aspect of your life. Whatever you are doing there on the last row, 3 students talking to each other, I can sense them, they are not listening to me at all. You तीन बैठे पीछे को देखो, last में. They are not listening to me. This is the scenario. I tell you, the time will come within 2-3 years, you will be having the classrooms AI enabled. And in that, our computer science engineers will play a very important role, that what a student is doing in the class, whole data will be come out to the student, means the teacher. So this is the future of AI. Be cautious. All the best. Thank you very much. Jai Hind, Jai Bharat.
Thank you, sir, for setting the perfect stage for today's deliberations. Now we would like to invite Professor Dr. Manoj Kumar Mishra to kindly felicitate Honorable Vice Chancellor. Now we would like to invite Honorable Pro Chancellor, sir, to present his opening remarks on the event. Sir, please.
Good morning, all of you. Dr. Manish K. Chaurasia, Senior Principal Scientist, Central Drug Research Institute from Lucknow. Professor Dr. Sanyog Jain, HOD of Pharmaceuticals from Naipur, Mohali. Dr. R.S. Tomar, officiating Vice Chancellor of the University. Dr. Manoj Mishra, the Director of AIP, and the Deputy Director, Dr. Mukesh, all the HOIs, HODs who are sitting here today, and all the participants of the conference, some of them online, and my dear students who are attending this conference today. The topic, artificial intelligence in drug delivery and design, from innovation to implementation. A lot has been spoken about artificial intelligence, but remember two words throughout your life: that artificial intelligence is neither artificial nor intelligent. Why? It is not artificial because it is real, it is existing. And it is not intelligent because from where it is drawing intelligence? It is drawing its intelligence from the data. And who has made that data? Human beings. Therefore, the data which has been made by human beings, from that the intelligence is being drawn. How can that intelligence be better than human intelligence? It cannot be. So if you remember these two things, you will not go drastically wrong. Right. Now, coming to artificial intelligence, the data which is there, from where it collects the data? It collects the data from, in case of drugs, from various hospitals, from various doctors, from the primary sources which are there on the government websites, from the sources which are there on WHO website, from the sources which are there on CDRI website, or on the other websites which are there, the government organizations, the NIPERS, so many academic institutions. Even today's conference will generate data. Today's conference, we have received more than 100 papers, for which we have got abstracts. For full papers, only abstracts, and for the posters, we have all this data is continuously being generated. So the quality of the intelligence will be as good as the data that is being generated. And you know, in academics, also in scientific research, you will be surprised. There is so much of fraud which is happening. It is also there. You can't deny that. There are fake papers which are being published today. There are papers which have been Scopus indexed and they are being retracted by the journals. That is also part of data which has gone in. So intelligence is also coming from those papers which are later being retracted. So all that is happening. So therefore, to rely too much on artificial intelligence alone will not be a correct thing to do, because AI itself is new. How old is AI? How old is AI? Not more than 10, 15 years old. So anything which is that new cannot be totally depended upon for all types of design. It can be an instrument. It is an instrument, no doubt about it, which can facilitate so many things. What is generative AI? Generative AI can generate 4 things. Number 1, it can generate text messages. It can generate text. Any speech you want to give or any question that you ask, it can give you a text output. On any question that you ask, it tries to give you an output which is made from the data. And then it is learning, machine learning. Also, it is happening from the new data which is coming up. The answers, anything you type today, the answer will be slightly different than the same thing if you ask the AI after 15 days. The answer may not be same, it will be slightly different because it is learning from the data which is already available, which is being continuously upgraded. So one is text. Second thing it can create is pictures. Like AI keeps asking, show me the quality of life on Mars if it exists. You just type there, it will show you. Show me the human beings if they settle down on moon, it will show you some pictures. Right or wrong, but it will show you. Then when you say, no, this picture is not correct, that the shadow is shown, the human beings cannot live on that. So it will then maybe try and refine and try and give some answers. The third thing it can do is generate formulas and algorithms. You tell him, please give me a small algorithm to arrange the 11 or the 100 figures in the ascending order. In C+, it will give you an algorithm. So it can generate algorithms, it can generate formulas. You say that these are the chemical reactions, what will be the output? It can give you the answer, that which is already available from the text. And fourth thing, it can also generate music. Any music which you say from the already having the music which is there, it can generate those. It can generate new molecules, possible molecules which could be used for drugs. It can generate those. it can generate from various reactions what are the likely outputs which can happen. So those are the things which AI can generate, and it can generate much faster than a human being would require, because human brain is not as good in processing data as the computers are, because the computers process data at a much faster rate, and that is the reason that why we are so much dependent on computers. But whereas the actual intelligence lies only in human beings. Any machine which is being produced by human beings, how can you assume that machine to be more superior to you? It cannot happen. But still, it has got the power of saving time, right? So those powers must be correctly understood and they must be correctly utilized. It is like this, like a Aladdin ka chirag. If you got, suppose you got a genie with you who can come out and do any of your wishes, but that genie should not be so powerful that he can tomorrow capture you alone and try to make you a slave. That is something which should not happen, right? Now, coming to the drug delivery, today's topic regarding innovation to implementation. The nuances of target identification, how the AI can generate molecules, how the virtual screening can happen has already been spoken. Now, the major challenges today are, number one, the quality of the data itself, which is suspect, and from where the data is being generated, the manner in which it is being generated, and manner in which the inferences are being drawn. Second thing, which is most important, is the interpretability. Now, the same AI which gives you something, if you interpret it, there are different people who will interpret the output of the AI in a different manner. So it is the interpretability which matters more than getting the right output from the AI itself. So for which human brains are required. And the third thing is the quality control. Of course, AI can just give you on paper. It cannot physically generate the drug for you and give you. Right? That no machine can do. So whatever the output given by that, from there, when it comes to producing the drugs actually, and the delivery systems, and the sensors to test the efficacy of that drug on human beings, using artificial intelligence. And it is that which is most important, because after all, a drug is to be administered on human beings. Then also regulations. Now there are regulations which are already existing, that how much time it will take for the drug to be administered, first on animals, then on say large section of or small section of human beings, then on a larger section of human beings, and then passed by various bodies, and then finally come into production. And then that also beta version first, and then coming to the actual version. All that process, it takes about 10 years today for a new drug to be developed and to come to the market. But as you are aware, during the COVID, the delivery was fast processed because of the urgency which was there. And still today, some of them are still saying that some of the drugs, the viruses, the injections which were developed, they may have a side effect which we don't know, or which may be seen only after a long time. So therefore, the regulations which are there, regulatory, that those will have to be modified if you are to depend upon the AI processes totally. That itself is quite challenging, that how AI developed design and how AI developed drug delivery systems can be implemented, and what changes will have to be made in the regulations. So that is something also which needs to be studied. So all those who are studying this topic must study the regulations. They must study the processes which are involved and the environment in which the drug is being made. So last time also on a similar topic when I had spoken, I had told 5 P's to be kept in mind, that is processes, products, the premises, environment, the process of quality control and quality assurance. So all those things need to be done deliberately. There, whatever time is to be taken, that time it should take. And to hurry up the production of drugs should not be the process, should not be the aim today, because the aim of some of the companies which are investing money into the drug industry is also to manipulate the data in such a way that the formula given by them will be shown as correct by the AI. A simple rule: on Facebook, there is a post which you want to get it removed. So 500 or 600 of you join hands and you say, this is a wrong post, please remove it. This is a wrong post. That post may be correct. Facebook will remove it within 20 minutes. What was uploaded was correct. But since 500 or 600 of you within a short span of time are telling that it's wrong, Facebook will take that this is wrong. They will remove it. This is the power of the social media, and this is the power of the artificial intelligence. Therefore, things which are wrong also can be made correct by inputting wrong data. That can happen. You want to sell a product, ask a class, a group to search for that product on the net. That product will become famous because there are so many people searching for it. It may be a useless product, but that will become famous because there are so many people searching for it. So the Facebook or the Amazon will start throwing at you that you search so-and-so product, that product will start selling better, which may not be the correct product. So all these things are happening, and by use of, there are influencers who are doing all this. And it is the wrong following. Sometimes we all tend to follow the wrong people and we get entrapped in wrong ways. After all, how are digital arrests happening? How are frauds happening? Because we follow the wrong persons. It is not that we want to follow, but the data manipulation is such that they make you follow the wrong person. Similarly, here also, the drug design, the drug delivery and all that can also be manipulated if those things can be manipulated. So this is one thing from which we all need to be cautious about, and we all need to go with one foot on the ground. Don't leave both feet on the ground and jump for AI. Whatever AI has told you, that is correct. There are instances when the mathematical formulas given by AI have been proved wrong. It is given wrong answers also. So remember, AI itself is learning. It is not refined to that extent. Although a time may come, maybe after a few years, when AI would have been much refined, AI would have learned the technique of rejecting the wrong data. AI would have learned of rejecting the false people who are trying to get things done like this, but that will take time. Until then, we have to tread with caution as far as AI is concerned. While using the importance of AIC and not downgrading it also that it is showing all shit at us. No, some things are correct also. So that is what I have to say. You got experts who will be talking on the subject of AI, drug delivery and design and innovation to implementation, how we can go. But a lot of thought needs to be done by the pharmacists who are sitting here, by the students of Institution of Biotechnology, and those who are involved in anything that is got to do with life sciences and with human beings and with animals also. So thank you very much. Pay attention to whatever is being said. And Jai Hind, Jai Bharat.
Thank you, sir, for your highly motivating and welcoming words.
We are now honored to welcome our second resource person, Dr. Sanyog Jain, Professor and Head of Pharmaceutics at Naipur Mohali, Dr. Sanyog Jain, a distinguished professor and head of pharmaceutics at Naipur Mohali, stands as a prominent figure in the field of nanomedicine and drug delivery. His career, marked by innovation and impactful research, began at DRDO, laying a strong foundation for his current academic and research pursuits. His primary research focus lies in developing advanced nanodrug delivery systems, addressing critical biomedical applications such as targeted cancer therapy, oral vaccine delivery, and gene therapy. This work has resulted in 14 granted patents, including the commercially significant tamoxifen SEDDS technology. Demonstrating his commitment to translating research into practical solutions, Dr. Jain's scholarly contributions are extensive, with over 250 high-impact publications and an H index of 67, reflecting his significant influence in the scientific community. His excellence has been recognized through numerous prestigious awards, including AAPS Fellow, and his consistent ranking among the world's top 2% scientists. Beyond research, Dr. Jain is a dedicated mentor, having guided numerous postgraduate and doctoral students. His extensive global presentations and substantial research funding further underscore his leadership and impact.
Now we request Dr. Sanyog Jain to kindly take over the session. Sir, please.
सबमिट कर लो, सबमिट कर देता हूँ। Honorable dignitaries on the dais, Dr. Tomar, Dr. Sharma, my senior Dr. Chaurasia, and all the colleagues. A very good morning to everyone, and thank you very much for a kind invitation. So I will give a brief overview of this artificial intelligence in drug discovery and drug delivery. So artificial intelligence, it is nothing but it is the ability of the machine to simulate the human intelligence. So based on the data, so it can analyze the data and it helps in taking the fast and accurate decision. So there are two components of this artificial intelligence, that is based on the machine learning and the deep learning. So machine learning, it is actually the machine analyze the data. So we need to input some data. So based on that data, whatever the data we are feeding in the machine, so it identify the patterns and it analyze the data and it then gives the decision. Deep learning, it is again the another type of machine learning algorithm, so that utilize that analyze the data based on the artificial neural network. Okay, so it actually mimics the human intelligence. So these are some real life examples of this artificial intelligence, like self-driving cars. Okay, this Boston Dynamics and this SEMO, these are the robots are there. Navigation system, which we generally use nowadays. Okay, chatbots. Okay, these are the customer executives. So we need not to talk to customer executives, so we can just chat. Okay, with any system. Okay, computer versus human games, and so many other examples are there. Okay, so these are some other examples. Okay, like this chatbots. Okay, this human robots, self-driving cars, navigation system, even the planes. Okay, they are nowadays in autopilot mode, so we can drive the aeroplanes also. Okay, these are the routine examples. Okay, some other examples like a smart home, okay, smart watch, Okay, so in the smart home, okay, so they identify our needs, what program we like to watch on the television, what temperature, what humidity level, okay, we feel comfortable. So automatically it adjusts the things. Okay, you might have experienced these things. Okay, like if you just Google any particular news or anything, okay, like I can give my example. Okay, last year I wanted to travel to US for AAPS conference, so I simply searched, okay, the flight connectivity from Chandigarh to Salt Lake City. Okay, so almost for 15 days continuously, okay, whenever I just open the Google, it shows, okay, these are the flight option. I did not check again, but it was automatically it was showing these are the flight option you want, you might be interested. And it was also showing, suggesting these are the hotel option. Okay, so this is the kind of artificial intelligence. Okay, if you order some pizza from any restaurant, so you identify, you select one particular pizza, you want this particular pizza. So next time, whenever you log in into that site, so it will automatically suggest, okay, you wanted to search this, you wanted to search this particular brand. Okay, so these are the routine examples. Okay, so sometimes it is good, but sometimes it also interfere with our privacy. So these are the drawbacks. Okay, this was, I was telling the example of deep learning. Okay, like nowadays your mobile phones, okay, they can be locked or they can be unlocked based on your face recognition. Okay, so this is the example of deep learning or artificial neuronal network system. Okay, the computer games, okay, so the computer games versus human games, okay, these are also based on this. Okay, so there are some advantage and disadvantage of this artificial intelligence. Okay, advantage, like it doesn't get tired. Okay, it takes the rational decision, correct decision, faster decision. Okay, and it is selfless with no breaks. Okay, it has several application in the field of medical industry. Okay, that we are going to discuss. But at the same time, it has having some drawbacks, like it is costly. Okay, it leads to unemployment. Okay, lacks in creativity, and no improvement with experience until unless we update the database. Okay, so that is the our input variables. Okay, now coming to the main topic. Okay, that is the application of artificial intelligence in the pharmaceutical science. Okay, so it has application in almost every field of the pharmaceutical science, starting from the drug discovery. Okay, so like drug screening, drug design, drug repurposing. Okay, pharmaceutical product development, that is the your drug delivery. Okay, so like nanorobots for drug delivery, control insulin delivery, combination drug delivery, nanomedicine. Okay, it is application in the quality control, quality assurance also. Okay, drug synergism prediction, drug antagonist prediction. Okay, in the diagnostic. Okay, and all the clinical application. So in almost every field, it is having the tremendous application. Okay, so first start with this role of artificial intelligence in the field of drug discovery. Okay, so this AI, it revolutionized the field of drug discovery. Okay, so it identify the correct disease target. Okay, it identifies the 3D protein, 3D structure of the protein or of that particular target. Okay, it helps in screening the drug molecule. So traditional approaches, we are time consuming. Okay, and it was very costly. Okay, so these are the different stages of a drug discovery. So first they start with the target identification and validation, then screening and discovery of the lead molecule, then preclinical development, and finally the clinical development. Okay, so the first step is the identification of the drug target. Okay, so based on the genomic and proteomic data of the pathogen as well as of the disease patient. Okay, so the AI, okay, helps identifies the best possible protein target. So there can be n number of a protein target which can be used for the drug discovery purpose. Okay, that can bind with the drug molecule to be for the therapeutic purpose. So based on the data, okay, proteomic and genomics data, okay, so it identified the correct target, molecular target or protein target. Okay, so it fasten the process. It also predict the potential target and its relationship with the disease. Okay, so this is a very quick and fast process. So traditional approach, it is, as I mentioned, it is very time consuming. Okay, then the second step after identifying the potential target, okay, that is the protein structure which can be used as a target. So it identify the 3D structure of the protein. Okay, 3D structure of the protein, if we can identify, then subsequently we can design the drug molecule which will fit or that will correctly bind with that particular target. Okay, so AI based on this deep learning, feature extraction, structure sequence relationship, and enhanced accuracy. So these are the several AI tools, okay, which can be employed. Okay, so it predicts the protein structure, okay, 3D protein structure in fast and efficient way. Okay, so that accurately identify the potential drug target identification. Okay, there are some other applications besides the help of this AI in the field of drug design and drug screening. It also helps in drug repurposing. Okay, whatever the drug molecule is there, because we know the different targets, we also know the different like a 3D structure of the protein. So we can know, AI can predict if this particular drug molecule can bind with that particular or the other protein also. It can have the application in the treatment of other disease, that is known as drug repurposing. And lastly, it can also help in personalized medicine, design of the personalized medicine by analyzing the patient data to suggest which or particular drug would be best suited for the patient and what would be the accurate dose for that particular patient. So like if the patient is suffering from some other disease, like if the patient is like kidney compromised, it is kidney is failed, kidney is not working. Okay, so then AI can suggest if this particular drug will not be suitable for the patient, or we can reduce the dose of that particular drug. Okay, so after discovery of the particular target, identification of the target, okay, so the chemistry people, they start the designing the drug molecule. Okay, so it helps in prediction of the hit or lead molecule. Okay, so initially the chemistry based people, okay, so they design the molecules on computer. Okay, so they design almost 1,000 or a series of the compound, okay, like 1,000 molecule they design. Okay, so then they fit into the particular target. So that is known as the screening. So prediction of the hit or lead. Okay, so they identify out of these all these molecules, this is our target protein, which drug molecule will accurately bind or perfectly bind, so which would be more effective. Okay, so that also reduce the attrition rate, reduce the overall R&D cost. Okay, and it can also predict the bioactivity of the molecule. So based on the drug target interaction, so it can predict this particular lead, okay, would be more bioactive. Okay, so the chemistry people can synthesize that particular molecule, so they will not waste time in synthesizing all the drug molecules. So it significantly reduce the time as well as cost associated with the R&D. So these are the several drug target interactions or different AI tools. Okay, they are available, okay, commercially. Okay, that can be used for this drug discovery purpose. Okay, so all these drug discovery, okay, so drug screening, drug discovery. Okay, so it is a branch of pharmacy that is known as bioinformatics, or in the pharmacy field, it is known as pharmacoinformatics. Okay. After the drug molecule, it is designed or screened. Okay, it's a biological property and material property like absorption, distribution, metabolism, and excretion profile. Okay, like ADME profile, it is also equally important before it can be considered as drug molecule, because the whatever the lead molecule you have identified in the discovery stage, they can be bioactive. Okay, but if they do not have, they do not possess the adequate ADME profile or properties, as well as the physicochemical properties like solubility, permeability, lipophilicity, etc. Okay, they cannot be developed as a drug. Okay, so this AI tools, okay, they can also evaluate the ADME properties. Okay, so in a faster manner. Okay, so AI-based model, they provide rapid, accurate, and prediction of these property to enhance the process of drug development. Okay, so absorption, how the drug is absorbed. Okay, the distribution, how the drug is distributed throughout the body, whether it is crossing the blood-brain barrier also or not, what is the volume of distribution. Okay, whether it is going into the target organ or not. Okay, so and metabolism is how the drug is undergoing metabolism inside the body, whether the metabolic product they would be active or they would be inactive, what would be the half-life. So all these attributes are very much important in subsequent development of the drug. So AI-based model, they used different tools or software to predict all these property, ADME property, and lastly the toxicity prediction also it can do. Okay, AI also helps in clinical trials. Okay, like clinical trials, we need to recruit some patients also. Okay, so sometimes the patient, they hide some information, but if we are using the AI-based model, okay, so we can select the right patient. Okay, like there are several digital tools which provide information about the patient behavior, like it's their sleeping pattern, symptoms, pain, cognitive function. Okay, so it helps in identifying or the correct recruitment of the correct patient. Okay, so this AI Cure, which is a mobile application, it is already used by certain companies for clinical trials. Okay, so after evaluation of the ADME properties, it's a material property like physicochemical properties, like its lipophilicity, solubility, pKa, okay, acid dissociation constant, melting point, log P value. All these are also very important. Okay, so AI, based on the AI tools, so we can predict all these parameters accurately and very quickly. Okay. So this AI-based model, okay, solubility-based model, they can predict the solubility. Okay, sorry, this crystallography solubility-based model, so they predict the solubility. Okay, the permeability models or and this degradation models are also there. So they predict how the compounds degrade in the different environment condition. Okay, permeability-based models are also there. So they predict the permeability of the drug molecule. Okay, crystallography models, they are also available. So they predict how the drug is crystallized and what would be the polymorphic form of the drug after getting crystallization. Okay, so because different polymorphic form, they have different solubility or different dissolution behavior, they have different stability profile also. Okay, so this AI-based model, so they correctly predict all these properties. And lastly, the drug excipient compatibility testing. Okay, so based on the data available, okay, the AI-based model can also predict whether the drug molecule would be compatible with the excipient which we are using or not. Okay, so for all these prediction, okay, so these different AI-based tools, as I mentioned, like deep learning, so that utilize this neural network, random forest and support vector, SVM system, QSAR models, natural language processing, NLP model. So these are routinely available. Okay, so and the different softwares, they are also available. Okay, and next very important consideration with AI in the field of drug discovery and development, that is the PBPK modeling. Okay, physiological based pharmacokinetic modeling and simulation software. Okay, so you might be knowing about this compartment models. Okay, so like it is a central compartment. Okay, so where the entire body is considered as one compartment. So whenever you inject the drug or you take the drug, so the drug will distribute equally into the all organs. Okay, there is concept of two compartment also. Okay, so one is called central compartment and another is called peripheral compartment. Okay, so the organs which receive the good blood supply, they will be considered in the central compartment like liver, kidney, lung, and the other compartment like skin or this hair, okay, or the other compartment which do not receive the adequate supply, so they are considered as tissue compartment. Okay, but nowadays this physiological based pharmacokinetic model, Okay, they do not only consider the blood supply, they also consider the organ size. Okay, they also consider the presence of your transporters, enzymes which can metabolize the drug. Okay, and the physicochemical property of the drug. Okay, and the presence of protein. Okay, so that can significantly change the overall pharmacokinetic of the drug. So this PBPK modeling, it is also a very emerging field in the field of pharmaceutical science, especially in the process of drug discovery as well as drug delivery. Okay, so and besides this predicting the ADME profile using this PBK modeling. So this PBPK modeling, they also have the ability, okay, like if you are doing experiments, okay, you have data on animals, like you have the rat data. Okay, so it can translate, okay, the rat data into the human data. Okay, if in the rat, okay, uh, the particular absorption distribution or whatever the pharmacokinetic parameter it is, this particular value, this would be in the human, this value would be there. Okay, not only the like it also simulate like age, sex, weight, body composition, organ function, genetics. Okay, so all these can be utilized and integrated in the PBPK model. Okay, that is not possible with the conventional pharmacokinetic models. Okay, so the drug property used in the PBPK model include molecular weight, log P, PK, protein binding data, okay, blood plasma ratio, metabolism, permeability, solubility, transport mechanism, lipophilicity, etc. Okay, so the input variable for this PBPK modeling would be your the drug physicochemical properties like its solubility, permeability, protein binding. Okay, this in vitro metabolism, active transport. Okay, so it can simulate the data. Okay, so initially, if you have the rat data, it can translate into the human data also. Okay, so that is the beauty of this PBPK modeling. So it is a very emerging field nowadays. Okay, so a lot of softwares, they are available. Okay, so like it is based on, as I mentioned, the organism properties, so anatomy and physiology of the organs, physicochemical properties and formulation factor. Okay, so it can we can feed the information and it will give your final result. And it can be applicable for like a cross species extrapolation. Like if you have the rat data or rabbit data, it can translate into the human special population. Okay, like if the patient is suffering from particular disease or any other complication, drug, it can also identify the potential drug-drug interaction. Okay, so a lot of applications are there with this PBPK modeling. So there are several software, they are commercially available, okay, for this PBPK modeling. Okay, so all these are the application of the artificial intelligence. Okay, so these are some other software. So especially this GastroPlus software, it is more commonly used for this purpose, simulation software. Okay, and this is also having some application in the regulatory. Okay, so it is not like a regulatory bodies. Okay, FDA will not approve. So FDA also, okay, approve the data which are generated using this GastroPlus and some other software. Okay, now coming to this AI in the formulation development, that is your drug delivery. So again, we need to give some input data. Okay, and based on that, it gives the possible solution of the drug delivery. Okay, so in the field of drug delivery, okay, or formulation development, first thing we need to decide what would be the most suitable route of administration for this particular drug molecules. Of course, we prefer the oral route is the most easiest and convenient way, but there are some drugs, okay, which cannot be delivered through oral route. Okay, so AI based on the property of the drug, okay, physicochemical feature of the drug like lipophilicity, log P value, and some other parameter. So it can give option. You can develop the oral formulation, or you can develop the parenteral formulation, or you can develop the topical formulation. It can also predict the drug resistance. Okay, so like again, we need to give some information as an input variable. Okay, so it can give the information whether the drug is causing any resistance, especially the antimicrobial resistance, AMR, which is very important nowadays in the field of this antimicrobial therapy. Okay, it also identified the correct dose optimization. Okay, so especially in the case of dose optimization in compromised patient like HIV patient, okay, or the patient which are suffering from kidney function or liver function. Okay, so compromised metabolism or compromised metabolic degradation is there. So it can suggest what should be the correct dose so that the patient will not suffer from the toxicity problem. Okay, drug delivery system improvement. Okay, so again, we can give some formulation variable and physiological variable as the input variable, and it can give information about what would be the diffusivity, okay, or the mucoadhesiveness if you want to develop, okay, or what would be the release profile we can obtain. Okay, so there are some commercial examples are also there based on this artificial intelligence. Okay, so like optimizing the drug release from matrix tablet. Okay, so the company, they prepared the matrix tablet using HPMC, hydroxypropyl methyl cellulose, and a dextrin. Okay, so they optimize, okay, what would be the optimum concentration, okay, of these two excipient to get the desired release profile. Okay, so from the matrix tablet. So they use some software, okay, for this purpose. Okay, so similarly, we can also have information. Okay, like if you want to prepare some matrix tablet or this, like this is the example based on this hot melt extrusion. Okay, so amount of this excipient and the diameter and the length of the solid lipid extruded, which we got using the this hot melt extrusion, and it can identify. Okay, it can correlate these two factors, input variable, with the dissolution rate and the dissolution profile of the final formulation using the AI. Okay, this control insulin delivery pumps, these are commercially available. Okay, so this is based on the glucose sensor. So if the limited glucose level is there in the plasma, okay, so glucose sensors is available. Okay, so they detect, because glucose is converted into the glucuronic acid. Okay, so it based on the like AI tool, so it actually trigger the insulin release from the insulin pump. Okay, so the elevated glucose levels, okay, they were come down. Okay, the glucose is converted into the glycogen. Okay, because of this. Okay, in the fixed dose combination also, so these AI tools, they help in predicting whether this fixed dose combination would be effective or not. Okay, whether this fixed dose combination will have any compatibility issue, or whether this fixed dose combination will have some toxicity problem. Okay, so it also identify, okay, whether we can use this particular fixed dose combination or not. Okay, in the quality by design, QBD, and design of experiment, this is also a very important tool in the field of drug delivery. So this is again the AI based tool. Okay, because in the conventional approach of drug development, okay, initially by hit and trial, we optimize different variables. Okay, and that lead to source of variable. Okay, source of variability in the final product. Okay, we optimize a particular ratio and we cannot go beyond that particular ratio. Okay, so but if we change the API, if we change the excipient, or if we change the process, okay, then overall change, okay, the final product would be highly variable. So if we want to minimize this variation, then we need to apply this quality by design. Okay, so quality by design, it ensures the high level of assurance of product quality for patient. Okay, and more efficient regulatory oversight, and it is also efficiency and cost saving for the industry. So nowadays, this FDA or regulatory authorities, they have imposed, okay, like in any formulation development, any formulation, if it is developed by, okay, or optimized by the principle of QBD, then only it will be approved. Otherwise, it will not get approved. Okay, so if we compare the QBD approach with a conventional approach, okay, so in the conventional approach, okay, we develop the formulation, okay, and then the quality of the formulation is tested by simple quality control testing, routine quality control testing, like you do for the tablet, like disintegration, weight variation, dissolution, etc. But in the QBD approach, okay, from the start, okay, from the beginning, we inbuilt the quality into the final product. So quality is ensured into the product, okay, by the process, by design based on the scientific understanding. Okay, so specifications are designed based on the batch history, but here specifications are designed based on the product performance requirement. Okay, so based on our requirement, we set the product specification. So the conventional approach, it is considered as a frozen process which discourages the change, but the QBD based approach, it is considered as a flexible process, okay, within the design space, so that allows the continuous improvement. Okay, so here we get the design space. Design space means we can play or we can vary the component ratio. Okay, it is not a particular fixed ratio. We can get the complete design space. Okay, so this is the more robust, okay, process which is having application. And this QBD, as I mentioned, it is approved by regulatory authorities. It is also mentioned in the ICH guidelines. So ICH guidelines, we see the Q8, Q9, Q10, and the Q10, okay, sections. So they mention about this QBD based guidelines. So this is the integral part of the drug development. Okay, so this QBD based approach, it is having the scope of continuous improvement in the formulation, development, or drug delivery purpose. Okay, so in the QBD, first it identify the source of variability. Okay, so source of variability, they are identified using this fishbone diagram. Okay, so there are basically 6 different sources which can cause variability in the product or quality. Okay, so first is the main, okay, these are called 6 M's: main, method, okay, materials, your manufacturing process, okay, milieu, that is your environment, and lastly the measurements. Okay, so we need to identify what can be the source of variability. Okay, so initially when we start this QBT, first we need to define the quality target product profile, that is known as QTPP. Okay, so we identify the specification. Okay, these are the quality attributes which our final formulation should contain. Okay, then we identify the critical material attributes. Okay, and the critical process parameter, we control them. Okay, so in a nutshell, we see the target. Initially, we set our target. Okay, then we apply the design, and finally we implement. Okay, so these are different stages I mentioned. Okay, initially define the quality target product profile, QTPP, and the critical quality attributes of the formulation. Okay, and then you identify the critical material attributes. Okay, your excipient, your API. Okay, critical process parameter. Okay, like if you are preparing the tablet, then like you are using wet granulation, dry granulation, or direct compression, or using okay, which machine you are using. Okay, then you need to link all these steps, link your critical material attributes and process parameter with your critical quality attributes. Okay, then apply your design of experiment or a multivariant design. Okay, then you get the design space. Okay, based on this design space, okay, you can develop the final formulation, and you will have the scope for the continuous improvement. Okay, so initially, as I mentioned, okay, it is the initial scoping. Once you identify your product, okay, product characteristics. So based on that, you define your quality target QTPP, quality target product profile. Okay, then you design the process. Okay, you optimize the process, and then you can do the large scale manufacturing. Okay, so the components of the quality target product profile. Okay, so they can be the critical and the non-critical components. So components that are related to safety, efficacy, identity, and purity and potency of the drug. So these will constitute the QTPP. Okay, so yeah, okay, no problem. Okay, so critical can be your assay, content uniformity. Non-critical can be your just appearance. Okay, and so these are the example of some immediate release products. So these can be your quality target product profile. Okay, so in this case, you can see this appearance, it is a critical one. Okay, but other are the critical parameters. Okay, there can be some other requirement for some specialized tablets, like if it is a scored tablet, okay, जिसको बीच में से काट देते हैं. Okay, sometimes the doctor prescribe the half tablet to be given. Okay, so in the tablet there is a like a marking system is there, so from where that can be tablet can be cut into the two halves. Okay, so the weight variation between the two halves, dissolution of the half tablet, it can also significantly affected if it is some specialized tablet like oral disintegrating tablets, like its hardness, disintegration. Okay, and if it is the control or extended release product, okay, then if the patient is consumed the alcohol, okay, so alcohol can dissolve most of the like coating. Okay, so alcohol induced dose dumping, so these are the additional requirement. Okay, so similarly we can define this CQA critical quality attributes. Okay, so in this case we can see, okay, the quality target product profile, and some can be, okay, critical quality attributes like your assay impurities, okay, content uniformity and dissolution, which can overall affect the performance. So these can be considered as critical quality attributes. Okay, so they can be linked. Okay, and last I want to highlight on this AI and 3D printing and 3D bioprinting. Okay, so this 3D printing is also a very emerging area in the field, modern field of medical field. Okay, so I will not escape this historical. Okay, so initial, like you just design anything. Okay, so this is just some example. Okay, but you can design your delivery system or a dosage form also, and you can actually prepare it using 3D printing. Okay, so there are three components like modeling, printing, and lastly the finishing. So modeling, just a virtual blueprints. Okay, so on computer you can design what you want to prepare. Okay, then actually you can do the printing, and lastly the finishing part. Okay, so modeling you just design on the computer. Okay, the printing, okay, so using the layer by layer deposition. Okay, so you can actually print, and lastly the finishing. So finishing actually after the 3D printing, so you can get this type of product, but after finishing you get this, like, so it is a kind of polishing step. Okay, so this 3D printing is also having application in the medical field. Okay, so it can design so many delivery system. It can also print, okay, or develop various organs also, like aortic, heart valves, blood vessels. Okay, artificial blood vessels, they can be developed in the future. Okay, we anticipate like different body organs like artificial kidney, heart, they can also be developed using this. Okay, and AI also helps some application in supply chain management. So I will just escape because time is over. Okay, but many pharmaceutical companies, they are using like this Merck and Pfizer. Okay, they use this AI based tool, okay, to forecast, okay, the demand and supply of this COVID-19 vaccine distribution. Okay, so it is also having application in the pharmacovigilance, drug interaction. Okay, so a lot of applications are there. Okay, so finally I come to conclusion. So this AI is making daily routine work of human easy and interesting by providing assistance in the field of traveling, manufacturing, research, and financial investment. AI also plays vital role in pharmaceutical world as it can be used for drug discovery, drug development, and drug delivery. AI can help pharmaceutical formulation scientists in designing the dosage form, defining the drug combination, and determining the drug pharmacokinetics. AI tools such as QBD and GastroPlus may reduce the drug development time and cut expenditure on the drug discovery and drug development. It also reduces the burden on clinical trials and provide better insights for drug development. And lastly, these tools will help in assessing the various contributory factors and their effect on drug quality, and ultimately transform the pharmaceutical industries to be more patient-centric. So thank you very much for your kind attention. So if you have question and time permits, then I will be happy to answer some questions. Thank you.
Anyone, you can raise the questions.
So I have given a very brief overview of, because time was limited, so I could not cover anything in too much detail. So I've just given the brief overview. AI can do so many things in the field of drug discovery and drug delivery.
Very good afternoon, sir. This is Dr. Alok Soni from School of Pharmacy, IITM University, Gwalior. Sir, I have one questions, or it may be doubt, that since AI is very vast application in drug discovery, so what about the application of AI in traditional medicine application? So what is the role of, means how we can involve the neural network or network pharmacology discovering the or preserving our traditional medicine system?
So traditional medicine, I think you are talking about the herbal medicine. Okay. Yeah, so traditional medicine, okay, so nowadays people are doing, so they are isolating or extracting the active constituent, they are identifying the active constituent, and they are making derivative of the active constituent to be more effective medicine. Okay, because in the herbal drugs, okay, lot of constituents are there. Okay, so some are effective, some are non-effective, okay, some cause toxicity also. Okay, so people are doing the extraction first. Okay, they are identifying this is the active constituent, and then they are developing the it's like using the AI tool. So they are developing the more effective derivatives or chemical compounds of, because most of the time lot of variability is there. Okay, like any example, haldi ka le lein hum. Okay, turmeric, curcumin. Okay, it is also well known nutraceutical. It is also used as a drug. Okay, but two different plants of haldi. Okay, if they are grown in same state or different state, okay, or in different season, so lot of variability in the curcumin content would be there. Okay, so if we just rely on the extract, okay, which traditional medicine, okay, people do, sometimes we get good efficacy, sometimes we do not get any efficacy. Okay, so the AI is doing, so already scientists who are working in the field of this traditional medicine, so they are doing this work. Okay, so with the help of AI, okay, they can formulate the more effective medicine, okay, based on the information. Yeah.
So I think this target prediction of protein binding
Yes.
efficacy, we can extrapolate or
Yes.
look forward for how this particular extract from the admixture
Yes.
can bind or stimulate or
Yes.
can do the pharmacological action.
Yes. Okay, so not only they isolate the active compound, okay, as I mentioned, they also make derivative, okay, further chemical modification of that compound, so that it can have the best efficacy, which the parent compound may not have. Okay. Yeah, thank you.
We extend our sincere gratitude, sir. The session was highly enlightening. It is now time that we proceed towards the release of conference proceedings with the selected papers screened out for the conference. We would like to request the dignitaries and members of scientific committee to kindly come forward for the release of conference proceedings.
Sharing the law. Is people sharing the law? Zoom time. बंद करो सर, फिर बंद करो किसी भी Zoom का करता
Amity Institute of Pharmacy takes immense pride to release major reference book entitled Nanotechnology-Based Strategies for Cancer Immunotherapy Concepts, Designs, and Clinical Applications, published by Springer Nature. Now, I would like to invite editors of the book, Dr. Rajiv Sharma, Dr. Vikas Pandey, and Professor Dr. Neeraj Mishra to join the release of book. Thank you, dignitaries. It's my privilege to introduce our chief guest, Dr. Manish Kumar Chaurasia, Senior Principal Scientist, CDRI, Lucknow.
Dr. Manish Kumar Chaurasia is a senior principal scientist in pharmaceutics and pharmacokinetics at CSIR, Central Drug Research Institute, Lucknow. He holds a PhD in pharmaceutical sciences from Dr. Hari Singh Gaur University, where he also earned his master's and bachelor's degree in pharmacy. His academic excellence is highlighted by numerous awards, including a university gold medal and recognition for outstanding research publications. His professional journey includes postdoctoral research at National University of Singapore and various scientific roles at CDRI. Culminating in his current senior position, Dr. Chaurasia's research focuses on pharmaceutics and pharmacokinetics, evidenced by his extensive publication record of over 105 papers, 2 dedicated books, and 25 book chapters. He also holds a patent and has secured 7 extramural research grants. As a dedicated mentor, he has supervised 13 doctoral and 65 master's degree projects. Dr. Chaurasia is a sought-after speaker, having delivered over 40 invited lectures. His contributions to the field are further acknowledged by prestigious awards like CRS Capsugel, Pfizer, and IDMA's Best Research Paper Awards. Now we request our chief guest, Dr. Manish Kumar Chaurasia, to kindly take over the session. Sir, please.
Thank you for the kind introduction. Good afternoon, everybody. Respected dignitaries on the dais, faculty members, students, ladies and gentlemen. Myself, Manish Chaurasia. I'm working as a senior principal scientist in CSIR CDRI Lucknow. I'll briefly talking about the role of artificial intelligence in drug discovery and development. So Dr. Jain has already talked about in detail about various AI tools which are used to develop in case of drug discovery as well as in formulation approaches. So I'll be talking about briefly the whole procedure or the guidelines or the protocols which has been laid down by our regulator for the development of the drugs, followed by 1 or 2 slides over the role of artificial intelligence in these drug discovery programs. So this is the content of my presentation. So drug discovery means we need to discover some molecules which has not been reported so far. So basically, drug discovery and development comprises of 2 steps. One is discovery of the molecules, or that comprises mainly of the synthesis of the molecules, and followed by conduct of various studies which are being laid down by the regulators so that that particular drug molecule can come into the market. This is the typical route of drug discovery. That's a very long and tedious process, and a lot of efforts are being generally laid down on this particular procedure. And you know, this particular whole process is very costly also. It is generally said that around $1 to $2 billion we need to invest in order to get at least one molecule into the market. This is being done majorly by, in US, it's being done majorly by big pharmaceutical companies, since you know the cost which is involved in the drug discovery process is huge. So not all companies or the small companies can invest that particular amount of the money, and they generally do not embark into the process of drug discovery or development. In CDRI, we are a public funded organizations. We are funded by the central government. So we are being mandated to work in the area of drug discovery development. So what is done in case of, you know, drug development, there's a long chain that starts from the synthesis of the molecules. So CDRI is one kind of unique lab we have, where we have all the regions starting from the synthesis of the molecules till clinical trials. You know, this long chain, and you need to align yourself at least in this particular whole process. So being, since I have a basic degree in the pharmacy, that's I am a pharmacist. So my area or my job in this particular whole chain is that whenever a molecule is being synthesized, that comes to us, and then we need to check the purity of that particular molecule. So what we do, we develop a method on HPLC for the quantification of that particular molecules, and then we check the purity of the molecules. Apart from this one, the regulatory guideline says that we need to make a representative formulation. Maybe, so generally what is being done, we prepare either a tablet or capsule formulations, then we have to assess the stability of these particular formulations. Apart from this one, we need to check the stability of API also, or the prospective API as per ICH guidelines. So, you know, this particular slide shows the steps or the typical steps which are involved in the process of drug discovery and development. So what I tell you, what is being done practically in CDRI. So, you know, we have divisions like, for example, the biggest division is medicinal and process chemistry division. And the responsibility of the scientists work in that particular division is to synthesize newer molecules. So what is being done, the scientists from this MPC division, they collaborate with the biologist. You know, we have divisions like cancer biology, parasitology, neurobiology, and then cardiovascular diseases. And apart from this variety of the regions, we have, for example, a scientist who works or develop new molecules in the area of cancer. So he generally collaborates with some cancer biologists. They sit together and they decide which target to pursue for the drug discovery. For example, you know, first step is to identification of a target. Target means basically a protein which is, for example, in case of certain proteins are there which the overexpression with that particular protein causes some specific cancer. So what is being done? You need to downgrade, or you need to synthesize some particular molecules which can interact with that particular protein, and then downregulate or reduce that particular protein, or stop that production of that particular protein, so that that can produce the anti-proliferative or the anti-cancer effect. For example, HDAC inhibitors are there in the market. PARP2 inhibitors are there. You know, in certain cancers, PARP2 protein or the label is being elevated. So we develop some inhibitors, we develop some molecules which can invert these particular PARP2 proteins, and that can, these particular molecules can be developed as PARP2 inhibitors. So the key steps involved, obviously the synthesis of the molecules. So the molecules are being synthesized by the chemist, and the chemist actually send these particular molecules to the biologist. And the role of the biologist is to check the efficacy of the compound. So the efficacy can be done by either the in vitro route or by the in vivo. In vitro means maybe with the help of some cell lines. For example, if you work in the area of development of drugs which are used for the treatment of breast cancer, then these biologists, what is being done? A chemist synthesize, say 50, 60 or 100 molecules, they send it to the chemist. Chemist apply some in vitro assay or MTT assay. With the MTT assay, they can ascertain whether these particular molecules is having some anti-cancer efficacy, and that is generally being measured in terms of estimation of the IC50 of the molecules. And we have critical guidelines also. If the IC50 of these particular compounds or the new molecules comes below 10 micromolar, then we consider them as a hit molecules. Okay. So if the activity is about say 20, 30 or 50 micromolars, obviously we'll have to discard these particular molecules. For example, if 1 or 2 molecules shows decisivity below 10 micromolar, then what is being done? Then they repeat the activity of that particular molecule through this again in vitro assay. Once the activity is confirmed, then they send us to pharmaceutics and pharmacokinetics division, where our duty is to check the purity of the molecules. So what we do, we develop a HPLC method for these particular molecules and then check the purity of the compound. If the purity of compound, we have obviously set guidelines for that also, that purity needs to be at least say 97 to 98%. Sometimes, the purity comes around say 90%. So what's the problem? The purity is very low. You are not sure whether the activity which is being exerted by that particular molecule is being contributed by the compound itself or by the impurity itself. For example, if the purity is 90%, that shows that the compound is only 90%, rest 10% is impurity. So are you sure? Can you say it for the sure whether this particular activity which is being given or shown by the compound is being contributed by the molecule or by the impurity? So what we do, if the purity comes below certain range, we send that particular molecules back to the chemist, and the chemist have various tools. For example, they have column chromatography. They run that particular compound with the column chromatography so that the purity of the compound can be enhanced. Then that particular compound comes back to us, and we again recheck the purity of the compound. And if that goes above that particular certain limit, we certify or we issue the COA or the certificate of analysis of that compound, and the compound goes back again to the biologist, and biologist reascertain the activity of the compound. Okay, if that comes okay, then that followed that particular molecule. For example, from a hit to lead. Hit means the compounds which have prospective activity. Lead means the chances of being drug or being druggable compound is high. So this particular process is known as hit to lead movement. Okay. So once that particular compound actually we subject them to in vivo efficacy. For example, in case of cancer, we have say 41 induced xenograft cancer model. We check the tumor regression efficacy of that particular compound. If that goes well, then that particular compound, for example, is being known as a lead compound. So that is identification of lead. This particular step, third one is identification of lead. Then the rest one is lead optimization. Lead optimization is basically a process I'll tell you in detail. This is a typical process which is involved in the synthesis of the molecules. So lead compound is a compound from a series of related compounds that is sum of desirable biological activity. So this molecule can be characterized and modified to produce another molecule with a better profile of wanted properties to unwanted side effects. Sometimes what happens, a compound which produces lead that has say some undesirable physicochemical properties, that has very low water solubility. If the compound is very low water soluble or log P of that compound is more than 5, sometimes the log P comes around 7 to 8. It means that the compound is very hydrophobic in nature. So it would be very difficult for us to get the oral viability with that particular compound. Second, so lead optimization is a process whereby the chemist optimize that particular with reference to introduction of some chemical groups so that the solubility of that compound can be increased, or the interaction of the compound with the target protein can be reduced. This whole step is known as lead optimization. You are optimizing your final compound. So once you get that optimized lead, then we subject them, that particular molecule, to various studies. So these are the properties of the good lead. You might have studied the Lipinski's 5 rule. What that says that the compound should have hydrogen donors less than 5, molecular weight less than 500 daltons, and the partition coefficient of the log P less than 5. But in general, what we prefer, that the partition coefficient of the compound should be less than 3 or around 3. That's better for any the prospective properties for or good properties for a prospective molecule. And they should not interfere with the P4-5 enzymes, okay, or the cytochrome P4-5 enzymes. And they should have some say reasonable water solubility, so that we'll get the good bioavailability with that particular compound. And they should exhibit cell membrane permeability. If your compound is highly soluble in water and that won't absorb through the GIT, then what's the issue of synthesis of that particular compound if that won't produce the good bioavailability? If the bioavailability of the compound is less, which shows that your compound will not enter into the systemic circulation and the patient will not get the good therapeutic response, or the dose of the compound has to be increased in order to get the optimum therapeutic response. and that should not exhibit cytotoxicity to the normal cells and should not be metabolized too rapidly. Now, these I'll talk about very briefly about the guidelines which needs to be followed in order to develop any molecules, and these are the guidelines which are being laid down by the regulators. In this case of drug development, in Indian context, the regulator is DCGI, or Drugs Controller General of India. So in order to develop any molecule, we have to subject them to these particular battery of the test. So these tests are being conducted. So what we do, we conduct all these particular experiments. The data has to be collected, and the data is to be collated in forms of a IND brochure, this investigational new drug brochure, or investigational new drug applications. So this IND brochure is being submitted to the DCGI, and the DCGI have a committee. They have Apex committee. They'll review the data, and they will call us for the presentation. So there are some queries. They grill us for the queries, and if there is some deficiency of the data, the application comes back to us that we need to provide so and so more data. We again generate the data and submit the applications. Once this IND application is being approved with the DCGI, we get approval for phase 1 clinical trial of that particular molecules. So in case of drug development, the major step or the major criteria not exactly is the efficacy of the compound, or rather is the safety of the compound. Efficacy may be compromised, but not the safety of the molecules. Safety means your compounds should be safe in nature. That should not be toxic in nature. Otherwise, what's the use of, or the rationale or use of efficacy if your compound is toxic in nature or compound is not safe in nature? It's the reason that they have provided various guidelines. We complete all these particular tests, then comes to us. And the major component is regulatory toxicology studies and the safety pharmacology studies, which shows the toxicity of that particular compound or the safety of the molecules. Apart from this one, we have to check the pharmacokinetics of the molecule also, and the pharmacokinetics may be in vitro also and the in vivo also. And the in vitro study include GI stability. You need to make sure that your compound is stable in the gastrointestinal tract. Otherwise, what happens? You administer that particular compound. If that compound degrades in the stomach, then what's the use? So what we do, we check the in vitro stability of the compound in the SGF, that's simulated gastric fluid and simulated intestinal fluid. And we also check the stability of the compound in the blood and the plasma. Otherwise, so we just want to make sure that your compound is stable in nature. We won't create much issue with the efficacy of the compound once this is being administered. Apart from this one, we need to check the CYP liability of the compound. So probably, I believe those students who have studied the subject of biopharmaceutics and pharmacokinetics, they know the importance of the CYP enzymes. So basically, these are the 6 or 7 CYP. We need to check the interaction or the liability of that particular compound. The major one is CYP 1A2 and CYP 2D6. These are two CYP enzymes. We have to check the liability. And what is being caused by this CYP actually enzymes are responsible for the metabolic activity of the compound. So if some enzymes are being overexpressed, okay, or some enzymes are being in the presence of the particular compound, so that has issues with the drug-drug interaction. Sometimes, the drug not only the single drug is being administered for any therapy, multiple drugs are to be administered. If one drug has liability on one CYP molecules, the level of that particular CYP molecules can elevate, and that elevation of that particular CYP can cause, say, the excessive metabolism of the other particular drug which is being concurrently administered with this particular drug. So CYP liability has to be checked. This is a regulatory requirement. Then, you have to make a representative formulation. You are not going to sell this particular API or the drug as a powder into the market. So a representative formulation has to be prepared, and a stable formulation has to be prepared. So what we do generally, we get the 3 batch of the prospective API molecules. And the batch has to be synthesized under GMP conditions. So we prepare representative formulations. Generally, we develop either capsule or a tablet formulation, and then we check the stability of the compound and triplicate following the ICH guidelines. And we also check the stability of the API also for 3 batches under ICH guidelines. Again, this is mandatory requirements. So ICH guidelines says that you have to assess the stability of the compound under 2 conditions. First one is accelerated condition. Under accelerated conditions, at 40 plus minus 2 degrees centigrade and 75 plus minus 5 RH, we keep this particular tablet or the API, and then we check the quantity of the compound or the assay of the compound on day 0, 1 month, 2 months, 3 months, 6 months. Okay, and if your compound is stable after 6 months, it generally gives a stability of 2 years. And then we keep under intermediate conditions at 30-75 degrees centigrade, and then again we quantify the compounds for 6 months, then 9 months, and then 12 months also. This is the major component of a drug development process: assessment of the safety or the investigation of the toxicity of the molecules. So every compound has to pass these particular tests, and the first test is genotoxicity of the molecules. We have to make sure that your compound is not genotoxic in nature. So generally 3 studies are being done. First one is in vitro studies, the AIM salmonella assay and the chromosome aberration assay. And second one, the in vivo studies include micronucleus assay or chromosome aberration, the rodent bone marrow. This is being performed. This is highly important. So generally what we do before moving ahead with say other tox studies, we subject it to 2 studies. One is genotox and other one is Herg channel liability. Herg channel liability is an assay which actually measures the cardiovascular toxicity of that particular molecules. These are the primary assay we have to conduct before embarking onto other tox studies. Since tox studies takes a lot of time. So the first tox study is single dose tox. So what is being done in case of single dose tox? This is being done in 2 species, 2 rodent species, generally being done with the rat and mice both. We take 3 elevated doses. For example, the ED compound, the effective dose of the compound is 50 mg. Then we subject it to first dose, the low dose is 50 mg per kg, 500 mg per kg, and up to 2 gram per kg. Okay, we administer to the 3 set of the animals. Then wait for the 14 days. After 14 days, then we record various parameters, then we sacrifice the animals and then check with the histopathology. We check whether there are issues with the toxicity of the molecules. And if this acute tox is okay, then move ahead with the repeated dose tox study. And the repeated dose tox is being done with either with the 14 days, 20 days, 60 days, 90 days, or 180 days. That depends on the the therapy duration of the intended therapeutic molecule. For example, the molecule which, for example, you synthesize molecule for say the cancer treatment, then obviously at least 1 to 2 months it has to be administered. So at least a 28 days repeat dose tox studies should be performed with that particular molecules. In case of molecules, for example, which has been used for the treatment of the malaria, then a 14 days repeat dose study is sufficient, since malaria therapy hardly of the dosage regimen is for 3 to 4 days only. So with this similar 3 doses level, we administer to the animals for up to 28 days. Then we record various parameters, including diet, weight, water content, and feed. Everything we record for 28 days. After 28 days, what do? We sacrifice the animals, and histopathology is being done, biochemistry is being done, just to check whether these particular compounds are safe in nature. So the repeat dose studies has to be conducted with two set of species. One is rodent and other one is rodent, non-rodent. And the non-rodent may include, say, a dog or a non-human primate like monkey. And these studies are very costly in nature. So it's routine for us that we work in the drug development, and then for the conduct of, since we do not have the dog facilities, we do have monkey facilities, but with the monkey facilities also the issue is with the procurement of the monkeys. That's a tedious process. Sometimes you don't get. So we outsource this particular dog study. The cost has been escalated 4 or 5 years back. We outsourced one study, one particular molecule that was used for as a rapid fracture healing agent for 80 lakhs. Now the cost is 3.5 to 4 crores for one dog tox studies. So we have one molecule in the advanced stage. We are conducting these studies, only left with the dog tox studies, and the quotation is more than 4 crores that we have to spend. Does this question come into mind, why we subject them to so much high doses? Just to rule out the adverse effect of the molecule. Sometimes, once that particular molecule comes into the market, so the doses are being administered at therapeutic dose only, but sometimes chronic doses are to be administered maybe 1 or 2 months. So we just wanted to make sure that even at the elevated doses, say 10 times doses, your molecule is safe in nature. And the purpose of, as per DCGI or OECD guidelines, the purpose is that the highest dose should produce some toxicity, but not the mortality in any case. But in the mid dose, they show some symptoms but no toxicity, and the lowest dose should be safe in nature. If you have some issues with the lower dose, then obviously your molecule should be dropped, and there should not be any mortality. If mortality comes, then your molecule is in danger. Apart from this doc studies, we conduct the safety pharmacology studies. 4 types of safety pharmacology studies are to be done as per regulatory guidelines. So these already I mentioned that we have to conduct the long-term as well as the accelerated stability studies with the with the API as well as with the representative formulations also. Apart from this one, photosensitivity studies or the photostability studies has to be conducted. If there is not much issue, if the compound is photo liable in nature, that's not an issue, then you store that particular molecules under amber color conditions, or you have to just protect the molecule. That's the reason. That's it. Otherwise, your molecule is not being thrown out of this particular drug development shelf. It is, say, photo-labile in nature. So this whole, formulation development, we have to conduct all these particular studies, including pre-formulation, formulation, and then post some packaging also. So everything has to be done, and the data has to be recorded. So once every studies are being done, we subject these, we prepare an IND dossier, and it is a huge document. And that whole process sometimes take 3-4 years to complete all the studies, then 3-4 years, if everything goes fine. So if you synthesize, say sometimes 1,000 molecules, 2,000 molecules, and the one molecule comes and as a candidate state, the molecule which is being subjected to the studies is known as candidate molecule. There's no more a lead now, that's a candidate molecule. Okay, so IND dossier is being submitted to the DCGI. DCGI then schedule a meeting with us, and then everything goes fine. Then we get approval for phase 1 clinical trials. So we have an experimental medicine and toxicology division where one scientist who is, MD in pharmacology, he takes care of this. Well, he looks after this clinical trials components. Currently, we are doing a phase 3 clinical trials with the phytopharmaceutical or Picolev or Picorizia curva for the treatment of non-alcoholic fatty liver disease. Apart from this, we already have phase 1 approval for two synthetic molecules also. So far, we have not initiated the clinical trials. We are looking for some partners. Being a government lab, we cannot sell these products direct to the market. So what is being done, once we develop these molecules, we try to license these particular molecules to any pharma company. And we always prefer, since the clinical trials takes a lot of money, that requires a lot of money. So what we generally require is that we collaborate with the companies so that the companies will also incur some expenditure which is being done on clinical trials. Apart from this, we knock the door of other funding agencies like ICMR also, since this phytopharmaceutical phase 3 trials is being funded by ICMR. So phase 1 studies are being done with the limited populations, and this is being conducted just to check the safety of the molecule or tolerability of the molecule. So it is being shared with the healthy molecules, not with the patients or volunteers. And phase 2 studies are being done with in case of patients or the non-healthy volunteers, just to check the efficacy of the molecules as well as the safety, or to optimize the dose of the molecules. And similar is the objective of the phase 3, but in case of phase 3 studies, larger population is to be involved. And one important point, in case of some chemotherapeutic of the toxic or the anti-cancer molecules, phase 1 studies are also conducted with the patients, not with the healthy volunteers. And other one is phase 4 studies. This is known as post-marketing surveillance. If any molecule comes into the market, that particular molecule is being, say, taken care for these particular adverse reactions. Generally, it is being handled by pharmacovigilance department. So since we do not have time, I'll not be talking about these phytopharmaceutical molecules, since these with plant molecules not now easy to develop these particular molecules. Now we have developed, our DCGI has developed these particular guidelines or released these particular guidelines, which is known as Phytopharmaceutical Act. So we have to develop this the plant molecules in this particular fashion so that they can be branded or they can be developed as a prescription medicine rather than a extracted one. So these are very harsh and tailored guidelines, so I'll not go much into details. Once we monitoring of these particular studies or conduct these particular studies are even tedious or harder than the synthetic molecules. And the phytopharmaceutical, the Picolev, what we have released or what we have subjected into clinical trials is a phytopharmaceutical only. So now I'll be talking only 2 to 5 minutes or where exactly is the role of AI in drug discovery and development. AI is nothing but a tool or the software which is used to accelerate any process. For example, if you wish to identify any particular target, so your drug may have what you assume that your particular molecule or a prospective molecule may have efficacy or interaction with 3 or 4 targets. So this particular whole process can be accelerated with the help of the AI, since AI is a tool which can perform the data very quickly. Otherwise, in the lab, we have to conduct the whole lot of assays or intro assays or high throughput assays so that we can come to the conclusion which target actually this particular molecule is interacting with. So with the AI, we have tools or the softwares and you can interact. Second one is, some of the library of various commercial libraries are available in the market. For example, a MABRAGE library is available. MABRAGE library has compounds in millions of the compounds. So I give you these 1 million compounds. Can you analyze these 1 million compounds? Even with the STS assay, that takes a lot of time. Is high throughput screening. You wish to check the in vitro activity of the compound, that takes a lot of time. So what is being done? Now again, we have AI-based softwares. What is being done? These particular softwares can screen all these compounds against the target. So which target for the target is one, the protein for which, or the protein against which you wish to develop any molecules. So these AI-based tool actually screen all these particular molecules and can give us the predictive or the, for example, say 10, 20 particular molecules based on the docking score or based on the some other algorithm outcome. So you get the compound, and these particular, say maybe 20, 25 compounds will have to subject them to say in vitro assays or laboratory experiments. That's feasible and that's possible. Otherwise, millions of the compounds, we cannot subject them to particular assays, or even if we do through this even with the high throughput screen, that will take a lot of time. Second, the lead optimization can also be done with the help of AI assays. We have a prospective structure of the molecules. We put the structure of the molecule in this particular software, and that particular software can give you some idea or the idea about the changes which are to be done on that particular molecule, so that you will get the say optimum therapeutic properties. For example, if a compound is say water insoluble in nature, what can be done? These particular AI software, or they can say that they can suggest some ways to include some specific groups or the side chains modification on that particular say head molecule or the lead molecule, so that you will get the optimized compounds. It's being done quickly with the help of these particular softwares. And then we have a software for ADMET prediction or absorption distribution methodology, actually, or the pharmacokinetic prediction software is also there. Dr. Jain has already explained in detail about all these particular softwares which are available in the market, including these ADMET softwares or the GastroPlus softwares. With the help of these particular softwares, offhand or the beforehand, we can get the information about the properties of the particular molecules, and beforehand we can synthesize or some more molecules, and based on a requirement and these particular we can move ahead with the optimized molecules rather than the one which we got earlier. And these AI softwares can also help us in clinical trial management optimization. These particular softwares can help us in selecting an optimum clinical trials. Clinical trials is a very complex process that takes a lot of time also since human volunteers involved. So we have to be very, very careful with these particular clinical trials. Doing an experiment on the animals is fine, but even if something goes wrong, that's not an issue. But the clinical trials, you have to be very, very cautious and careful. Nothing should go wrong in case of clinical trials. Otherwise, the sponsor of the clinical trial may be in trouble. So these particular, the design of studies, design of the trials can be very optimally done with the help of these particular AI softwares, including the inclusion and the exclusion criteria with the volumes which are to be selected in case of clinical trials. So these are other softwares like DeepCam, DeepTox. DeepTox is software which can predict the future the toxicity of these particular any molecules. Even if we get say some softwares which can predict the genotox or the hERG channel liability of the molecule, that's a great help for us. Last time we have one particular molecule that's the PCSK9 inhibitor, and we conducted variety of the studies on that particular molecules, including the acute tox, and then genotox, and then this hERG channel liability. But with the repeat dose studies or the dose range finding studies, that particular molecules were became toxic in nature. 1 minute. Then that became toxic in nature, and we had to drop that particular molecule. So we have to be very much cautious, since this whole process very tedious, and that involves a lot of money also, and then efforts also. So I believe that since we work in the area of drug discovery and development, and with the coming days, obviously we'll start involving these AI-based tools to a very large extent, so that the whole process of drug development can be accelerated and can be optimized. So I wish to acknowledge these various funding agencies which gives us fund to conduct these particular studies. So being part of the CDRI, one of my role is to work in the area of drug development. Second one is then being a scientist, I have my own interest in the area of nanobiotechnology or nanotechnology. So my students, since we supervise the PhD students, so we work in the area of nanotechnology, and in order to get funding, we have to tap the door of these funding agencies. So once we have the funding, then obviously our students can work for their PhD programs. So I wish to thank all these funding agencies. Thank you all for patience listening, and the forum is open. If you have any queries, these are my PhD students who've done. If you have any queries, free to ask. So I have actually briefly described everything. Otherwise, this whole drug discovery and development, that takes a lot of time. Thank you.
Thank you, sir. Taking the session forward, we would request our Honorable Pro-Chancellor to kindly felicitate our Chief Guest, Dr. Manish Kumar Chaurasia. Thank you, sir. Now, I would request our Honorable Vice Chancellor to kindly felicitate our keynote speaker, Dr. Sayok Jain. Thank you, sir.
I would request our Honorable Director to kindly felicitate our Honorable Pro-Chancellor, sir. So please. Thank you, sir. I would request our Honorable Deputy Director, sir, to kindly felicitate our Honorable Vice Chancellor, sir. Thank you, sir. We kindly request all the dignitaries, chief guests, keynote speakers, and faculty members to assemble on the stage for a group photograph.
Ladies and gentlemen, VIP members and dignitaries are requested to head towards VIP lounge B block for lunch. Delegates and students are requested to move towards room number 215 and 216, block C, to participate and attend the poster and oral presentations. Lunch break for delegates, students, faculties, and staff will start from 1:15 p.m. Room number 315 and foyer area, block C. Delegates and participants are requested to assemble at C Block Seminar Hall again at 2:15 p.m. sharp for the second technical session by respected Professor Dr. V. Ravichandran.
Thank you.
विजय साउंड नहीं आएगा जब तक तुम एडवांस में जाके साउंड शेयरिंग पे क्लिक करके फिर फाइल पे शेयर करोगे तब जाके आएगा। साउंड नहीं आ रहा अभी। ठीक है आ गया आ गया आ गया। हो गया विजय हो गया। हाँ, ठीक है शेयरिंग स्टॉप कर दो हो गया।
सर डन।
ओके
एडमिट करना क्या है?
एडमिट हम भी कर सकते हैं वी आर होस्ट ना।
तो एडमिट अपना कैमरा कहाँ गया?
क्या हो रहा है ये? रजिस्टर हो गया ना।
यहाँ से जो भी आप बोलेंगे जो बाइक बाहर जा रही है आवाज जो आपकी गेस्ट है या और कोई आपकी नहीं नहीं भाई
फार्मेसी वाले कोई जो है ना
तो उनकी क्या वो आई पीपीटी भी आई है।
नहीं वो कुछ शेयर करेगा तो कुछ शेयर हाँ उनका पीपीटी भी है अगर काम नहीं करेगा तो
है ना
एपी ऑन रहेगा।
कौन सी जगह वही तो इसमें ऑडियो भी है क्या?
नहीं
ऑडियो से क्या बोले थे आप चलेगा
वो बात करेगा तो सुनना है ना।
नहीं वो तो आ जाएगा जैसे अभी सर लोग बात कर रहे थे ना।
हाँ
वो तो
ये पीपीटी वो खुद शेयर करेगा अगर कुछ टेक्निकल
हाँ।
है तो हमको करना पड़ेगा।
सर चेक कर लिया हैंड ओवर मैं रहूंगा यार कुछ ऐसा कुछ किसी का है ना बोला नहीं था तो मैं
अरे आप मेरे सामने नहीं इतने बार कॉल किया हूँ। एक बार आप मैं आ रहा था रास्ते में था उस टाइम पे भी आप कॉल उठाते तो मेरे साथ या
मैं निकल मैं पहले भोजन करता हूँ ब्राह्मण कोई बात नहीं सर
पर आज थोड़ा सा
कोई बात नहीं
स्पेशल बनता है ना भी बनता है ये सोच रहे थे आप खाना खाना शुरू कर दिया है फोन नहीं उठा रहे
मैं खा ही रहा था क्यों पैंट टाइट था घंटी भी शायद देंगे आप मुझे देख दीजिएगा क्यों नहीं बजी घंटी और शायद तो बाइक पे होगा जो आपने फोन किया होगा 60 पार्टिसिपेंट ऐड हो गए हैं आपके ये रिजल्ट का फुल अच्छा ही है बोलना नहीं लड़का लड़की आते थे हाँ बोलना नहीं ये थोड़ा ध्यान रखना कोई ये आगे बढ़ गया या है ना आप चक्कर काटते रहिए ये 1 घंटे का प्रोग्राम है ना कितना है ये 1 घंटे का प्रोग्राम था एक ही कितने लोग हैं ये प्रोग्राम आज की जगह कल करते हैं या फिर
क्या हो गया कल भी आना बेकार से गया कल आपसे इंस्पेक्शन के टाइम पे भी बेकार से आना के लिए इंस्पेक्शन के लिए शनिवार के टाइम
हाँ सी आएगा
नंबर के लिए तो
उस दिन आधी आधी यूनिवर्सिटी आएगा HR IT
only the department background the other department supportive है
हेलो मैं पहुंचा तो
हाँ start. Yes, sir. अभी रविचंद्रन sir join हो गया.
Yes, joined.
Yes, he's over.
How many participants are there?
Now, so 57.
What about the waiting room?
Waiting room, I said him as enable until the meeting starts. वो ब्लॉक करके रखा है बट अभी enable कर रहे. Guests have joined. We now the meeting.
Now in the group, you can put a message in all the participant group.
Sir, मैं उसमें नहीं
There,
हूँ.
no, here.
मेरे को enable admin नहीं बनाया sir. That is what all faculty make students to assemble.
Okay, I'll check.
It's Manoj sir. Director sir. Director sir. Where is director? Manoj sir.
Yes.
यहाँ से कर सकता हूँ आ जाओ आओ इवनिंग कर लो आओ तो वही इसको है ना ऐसे एडजस्ट करना ठीक है हेलो ये लाइट जल्दी है क्या अब बोलो अब अगर आप हो जाए क्या करना है कॉन्टेक्ट में
They asked for questionnaire. Then we will call us. So keep your mind. There is no time. Please be serious. Keep your mind. We are going to start now.
Good afternoon, everyone. Everyone is asked to keep their mobile phone on silent mode. Please be seated at your positions. We'll be starting shortly.
The speaker is from Malaysia. He is a very good speaker.
Welcome all to the second technical session of the conference. Without further ado, let's dive straight into our technical session 2, where I, Shraddha,
And I, Harshdeep, would like to introduce you to our keynote speaker of the online session, Professor Dr. Ravichandran Veerasamy, who is joining us in online mode. Professor Dr. Ravichandran Veerasamy is a distinguished professor at AIMST University, Malaysia, where he leads the pharmaceutical chemistry units and serves as Deputy Director of Postgraduate Studies. With his 26 years of expertise, he excels in medicinal compound synthesis, phytochemical analysis, analytical method development, and molecular modeling. He also served as an elected Senate member, a prolific researcher. Professor Dr. Ravichandran has supported by the numerous universities and FRGS grants. He contributes significantly to academic publishing as an associate editor and editorial board member for multiple journals, as a reviewer for over 125 journals. His academic influence extends to PhD thesis adjudication for 16 universities. He is a highly cited scholar and has been recognized among the world's top 2% scientists by Stanford University for 5 consecutive years. He has mentored numerous graduate and postgraduate students, including PhD candidates, solidifying his role as a leading figure in pharmaceutical chemistry and analysis. Now we request Dr. Ravichandran Veerasamy to kindly take over the session. Sir, please.
Sir, please unmute, sir. Enable your
Okay.
video.
Okay.
Yes, your voice is audible. Please enable your video, sir.
Yeah, already enabled. You don't want the video, huh?
Yes, okay, sir.
Okay,
Slide is good.
okay, thank you.
Sir, you can start.
Okay, thank you. Thank you for the detailed introduction about me. First of all, I would like to thank the management of Amity University, Madhya Pradesh, for inviting me to deliver a talk on the international conference. And my special thanks to Dr. Mukesh, Dr. Neeraj, and Dr. Sagar for continuously contacting me about this talk. Then my thanks to the audience also. So the morning 2 session was over. I think my friends, Professor Dr. Sanyog Jain and Dr. Manish Saurasia has discussed about artificial intelligence in drug delivery system. So in my talk, I'm going to discuss about the artificial intelligence in drug design and discovery, the total whole process, how it is influencing the drug design and discovery process. So why we ought to go for the artificial intelligence? So in earlier years in the 19th century, our ancestors, they have used the conventional method, that is traditional method. They have used the plant extract and animal extract for the treatment. Then later on in the initial of 20th century, they started to isolate the active constituents from the plant extract. Then they have used for the treatment. Then after that, slowly they introduced the computer-aided drug design in the year of 1960s onwards. But everywhere, why we are going for the development slowly by a conventional method to the extraction method, then computational technology, now artificial intelligence. Yes, in drug design, as you all are aware, that it will take a lot of time. It is a time-consuming process. So you want to develop one drug, it will take minimum 12 to 15 years we have to spend, and the manpower and the cost-wise, they have to spend more than $800 million US dollars. Then the manpower, all these things you have to count to develop a one drugs. So they want to develop one drugs, at least minimum 1,000 to 10,000 compounds they have to prepare, and sometimes in that one compound may be active, sometimes all the 10,000 compounds are inactive. So that is the reason why it took long time. Then when they introduced the computer-aided drug design, it drastically reduced the time from 12 to 15 years to 8 to 10 years like that. But even though the 8 to 10 years, it's a long period. Yes, recently we faced in 2020, we faced the pandemic, that is COVID-19. So that time we know what we faced, whatever the facility we are having, the computational drug design, even the artificial intelligence was there. But even though we faced a lot of difficulty to find out the track within a short period of time, even though we managed, within 3 months, we have found out some of the already FDA approved drugs are useful in the treatment of COVID-19. That was possible only because of the computer-aided drug design and the use of artificial intelligence. Otherwise, we couldn't imagine that what will be the last. Yes, even though after using all these things, the death rate was very high. But if you are not having that time, all this technology, then you think about that what was the last. So nowadays, the artificial intelligence, not only in our pharmacy field, wherever, whatever the field, you can take any other specialization like any other like medicine, dental, or technology, engineering, wherever you go, without artificial intelligence, we cannot see anything. Because that is most essential part in our life, even though it is one of the factor which will reduce the record manpower. Because many of the people, they are losing their job because of this artificial intelligence. That is in the other part, but the main part is the speed and the output what we are getting from by the using of artificial intelligence, the result what we are getting, the accuracy of the result, that is better than the human when we are doing the same work. That is the advantage when we are using artificial intelligence. So artificial intelligence is transforming how to discover and develop new medicine. Means
Sir, your slide is not moving, sir.
no, I have not moved the slide. First slide only.
Sir, kindly move your videos.
Yeah. Now it is second slide. Can you see here? It's visible. Hello.
Your audio is coming, but video is not.
Hello. My second slide is visible or not?
Yes, sir.
Okay. Okay. Thank you. Okay, the computer-aided drug design. Just in the earlier, just now I told the conventional method, there is a lot of issue they faced. Then slowly in 1960 and 70 onwards, they introduced the computer-aided drug design in the drug design process. So because when we are having in the earlier in 19th century, we were not having that much data. So it was easy for the human being to manage the data. But in 20th century onwards, we are having a lot of data. It's not possible for the human to remember all those data to maintain the data. So we need, they have started using the computational technology to store the data and utilize, analyze the data and the use of the data. So that is computer-aided drug design to discover, design and optimize biological active compounds, means the drug. So the importance, it is drastically reduce the cost of the drug design process in pharmaceutical development. And the main thing is the virtual screening. That is the important development in computer-aided drug design. That is the virtual screening. Virtual screening means virtually we screen the compounds instead of what we are doing in the laboratory. We have to do the bioassay. For suppose we are having 1,000 compounds, we want to do the bioassay, we have to spend a lot and time consuming and chemicals, all these things. So instead of that, we can do all the things virtually by using docking or QSA or any one of the techniques, pharmacopoeial modeling, any one of the technique we can use virtually. By using the software, we can predict the activity, the biological activity. So the compounds which is not showing the activity, then no need to synthesize those compounds. Then later on, whatever the compound showing the affinity, then we can synthesize that compound and we can go for the biological activity. So it drastically reduces the number of the compounds to be synthesized. By conventional method, suppose if you are synthesizing 100 compounds, then you have to do the bioassay. Sometimes one compound will be active, sometimes that also will not be active. But in virtual screen, the computer-aided drug design, before synthesis itself, we can analyze the drug. We can draw the structure by using different software. Then we can do any one of the computational technology like QSR, 2D, QSR, 3D, QSR, CR, docking studies, molecular, and any one of the techniques we can utilize, and we can reduce the number of compounds to be synthesized. So just now I told in conventional method, we plan to synthesize 100 compounds, we can reduce the compound to 4 or 5. The active compound that we can synthesis and do the bioassay. So it drastically reduces the cost as well as the time and the manpower and many things. So the historical development evolved from simple modeling tools in the 1970s to sophisticated platform today because of the influence of artificial intelligence. So in traditional computer-aided approaches, there are two approaches. One is structure-based drug design, the another one is ligand-based drug design. So when we know the structure of the target means either the protein or the receptor which is responsible for the disease. So once we know the structure of that, then by using that, we can design the complement ligand, means the chemical compound which is having affinity against the particular target, the receptor or protein. So that is structure-based drug design. So for that, we need the 3D structure of the biological target. If we don't have the biological target, then we can substitute some other computational technology like homology modeling. We can design the target and do the structure-based drug design. Suppose both are not possible, then we have moved to ligand-based drug design. In ligand-based drug design, there is no need to know about the target, but we should have some number of compounds and their biological activity. We should know the biological activity. Suppose I want to do the ligand-based drug design for COVID-19, then I should have some 20-25 compounds of the same nucleus compounds with its anti-COVID activity. Then only I can do for the ligand-based drug design like QSAR, pharmacopoeial mapping, modeling. So by using this, we can develop the model. This is one of the best example for one of the artificial intelligence technique, that is machine learning. QSR is a type of machine learning techniques, because first we can train the machine to develop the model. Once you develop the model, by using that model, we can predict the activity of the unknown compounds. That is the concept in the QSR techniques. So where we are using the machine learning, and here also structure-based drug design also when we are going for virtual screening. So first you have to train the software, particularly for docking, we have to train the software. Then we can predict the biological activity of hundreds or thousands of compounds in a day. So that is also coming under a type of machine learning. So the limitation of traditional computer-aided methods, just now I told the time consuming. It's also a time consuming process because conventional it took 12 to 15 years means the computation when we are using the computer-aided drug design, it took near about 8 to 10 years, just 2 or 3 years lesser than the conventional method. So complete all the process. Then computationally expensive. That is the very important things because when we are doing QSR, we no need of the superpower computer or all other software tools. But when we are doing molecular dynamics, so we need more computational power like a superpower computer, and sometimes the molecular dynamic process also it will extend for 1 week, 1 month, more than that also. So that is the problem what we faced when we are using the computer-aided technology, particularly the structure-based drug design. Then the limited chemical space. The traditional method explore only the tiny fraction, means small fraction of possible drug-like molecules. That is the another drawback in the ligand-based drug design. So what the scientists did, they introduced AI in drug discovery. So for example, today I'm presenting the slide. You can see on the slide, usually before this my talk, today talk, near about I have given 70 to 75 talk, but every time I used to sit and prepare, I have to collect the information for 5 to 6 days, then the preparing slide for 2 to 3 days. What totally I used to spend 9 to 10 days to prepare the slide. But today, for this talk, I have prepared the slide within 20 minutes. How it was possible? Earlier, I used to spend near about 8 to 9 days to prepare the 30 to 40 slide for your talk. But for this talk, I have spent for the PowerPoint preparation, I have spent hardly 20 minutes. And the preparation, I have spent another 4 or 5 hours. So totally 6 hours I have completed the preparation. The reason is AI. Because I used the AI tool for preparing the slide. That is our tool is AI tool is Gamma. So when you are using Gamma, it has given all the information what we want to cover in that particular topic with beautiful diagram, means the related diagram and the point-based information, and it was making somewhat, I think, these beautiful slides. So the reason is AI. Because I thought earlier I also don't want to use the AI, but today I want to give a talk on this AI. So I tried to utilize that option, then only I understand. So instead of wasting 9 to 10 days, within 5 hours I got ready for the presentation. So that is the thing. So whatever the new technology, if you want to use it in the correct way, we can use it. That is the thing. So introduction to AI in drug discovery. So only the scientists also thought we can introduce the AI in drug discovery because already we introduced computer-aided drug design. Still, we are facing a lot of issues like time consuming, computationally expensive. So they brought AI. So once they brought AI, what is the advantage? The speed. When we are doing molecular dynamics or the docking studies for a month in a normal computer-aided drug design, when we are using AI, within a day it will be completed. We can complete the task. That is the speed. Then it's not needed the computationally. It's not that much expensive compared to the computer-aided drug design, and ability to explore vast chemical spaces beyond human capacity because it is going to think like human. So it give lot of information and lot of optimization is possible because there are different techniques are there in AI. So each and every technique is specific for some properties. So it will optimize all the properties together because as a human, when we want to do, suppose I want to do ADME studies, I have to concentrate on ADME studies. And then I have to go for synthesis, formulation one by one. Suppose I am following the computer-aided drug design. So there also the problem is that suppose I want to do the QSR studies, I have to do the QSR studies first. Then I have to move to docking studies, then ADME studies, and toxicity prediction studies. So one by one I have to do. So again, it is the time consuming process. But when you are implementing the artificial intelligence at a touch, it will do everything within very minimum means very short time. It will complete the task. So what is AI? Artificial intelligence. Everywhere we are using, even now, the automatic car, you can see there is autopilot is there. That is one of the AI technique in when you are going for some star restaurant, robot, they are doing the service. The server work it is doing by the robot. That is AI. Everywhere, wherever you go, everywhere the AI technology is there. So what is the AI? The definition is any system or any machine that mimic human intelligence, means that capable of the task, capable of performing the task which is performed by the human. So the machine or any system that will think like human, and it will give the suggestion or it will give the model to predict the property. So that is called as artificial intelligence system. It learning patterns and making prediction from data. Yes, all it is based on the old data, whatever we are having the databases. Based upon that only, all these artificial intelligence techniques are working. Then one of the technique is machine learning. So everything is based upon the algorithm, what algorithm we are using. So the algorithm will train the software to do the things. So in machine learning, algorithm that improve automatically through experience and data analysis. So these are the three types of AI what we are using in computer-aided drug design. The basic one is machine learning. Machine learning means first we have to train the machine. Then by the training, the machine will develop the model. Then we can use that model to prediction of the activity. The best example just now I told the QSR technique, quantitative structure activity relationship technique. That is one of the best example for machine learning. So first you have to develop the model by using the software. When we know some experimental data, by using that experimental data, you have to develop the model first, then validate, then we validate that model, we can use it for prediction of the unknown compound. So first we are training the machine, but that training, it will develop the model and we can use that model to improvise the all other properties. That is machine learning. In deep learning is the subset of machine learning. Machine learning is the subset of artificial intelligence. Artificial intelligence, it is a broad term, it is the umbrella term for covering all the things. So under artificial intelligence, the machine learning is there. Under machine learning, the more deeper is the deep learning. Deep learning is same like a subset of machine learning, means it is also using the data. By using the data, it is going to develop the model. But in machine learning, we have to train the machine. But in deep learning, it's not like that. It is going to use the algorithm, that is artificial neural network. So by using artificial neural network with multiple layers, the algorithm will train the software to develop the model by itself. That is the advantage of deep learning compared to machine learning. Machine learning, you have to train the machine, then it will develop the model. But in deep learning, the algorithm make the software to train by itself. That is the advantage of deep learning. The next one is reinforcement learning. Here, the optimization is the major process. It will optimize all the properties, whatever we are going to do. That is the most advanced technique. So in machine learning, in computer-aided design, in machine learning itself, there are different types are there: supervised learning, unsupervised learning, then semi-supervised learning. So by the name itself, we can understand supervised. Just now I told. So we have to train the machine. So for that, we should have some experimental data. By using that experimental data, we can train the machine. That is supervise the machine. Supervised learning. So once you trained, then you can use by the training, the machine will develop the model. Then by using that model, we can predict either it is a biological activity or toxicity or any other things. We can easily predict the things. That is supervised learning, means we are going to use the labeled data. Means we are having 20 compounds with whether it is active or inactive, then active, whether it is moderate or highly active or less active. All this information, the labeled information we need. That is supervised learning. Then unsupervised learning. In unsupervised learning, here it will identify the patterns without the label data. Just we have to give the information. So the machine itself, it will analyze. So it will segregate the things into different groups, different clusters. We can tell it is a clusters, means highly active, moderately active, less active. Then it will develop the model and do the prediction. That is unsupervised. Here we are not going to train the machine. Automatically it will divide according to the activity. That is unsupervised. So semi-supervised, no need to explain. It is the mixture of both supervised and unsupervised learning. Then deep learning and computer-aided drug design. So just now I told the basic algorithm for the deep learning is artificial neural network. So it will like how our brain will work, how the brain is attached to different nervous system and how it is working, the neuron system, then how it is working. So based upon that, they have developed the artificial neural networks. So it is contain multiple layer network that process complex molecular features. So that is the foundation of modern AI in drug discovery. So in that there are number of techniques. Here I have given two techniques. One is convolutional neural network. The second one is recurrent neural network. In convolutional neural network, the specialized for grid-like data such as 2D molecular representation, excellent for image-based molecular property prediction. Then recurrent neural network process sequential data like SMILE strings. So when we are drawing the structure, we can convert it into SMILE form. So by using that SMILE form, it will generate the predict the properties. So ideal for generating new molecular structure. The next one is reinforcement learning in computer-aided drug design. That is the most advanced technique. It uses agents that learn optimal action through rewards. That only I told. It will optimize all the properties at a time. It's not like one by one, like in machine learning or deep learning. It will optimize all the properties. So optimize exploration of chemical space with exploitation of known good properties. Suppose I'm having some data. So I want the data for anti-cancer activity of some compounds. I want to improve the anti-cancer activity. So when we are using the reinforcement techniques, no need to do one by one. So automatically it will find out what are the properties responsible for the anti-cancer activity. Then it will give the suggestion which one we want to improve. So all the information we can get. That is the core principle of reinforcement learning. Then drug design application iteratively modifies molecule to optimize multiple properties simultaneously. That is the advantage of reinforcement learning. It mimics medicinal chemistry decision making. So how your medicinal chemistry we can decide when we want to develop your design your new drugs. You have to think many things from the raw material, what is the pre-formulation studies, how we are going to synthesize, how we are going to formulate, how we are going to do the in vitro studies, in vivo studies, preclinical studies, all the things. And if the synthesis is not possible, then how to change the synthetic path, the methods, all these things combined together, it will optimized by the reinforcement learning. Then the success stories, one of the examples is DeepMind AlphaFold, which is used to predict the protein model. So when we are not having the protein structure, so by using this DeepMind AlphaFold, we can easily get the structure of the protein, which is the protein structure of the X-ray crystallographic structure of protein or NMR structure of protein or the electron microscopic structure of protein is not available. Then how to do? So that time we can use this DeepMind AlphaFold reinforcement learning method to create the unknown protein. Okay, means the unknown, the structure is unknown, but the sequence is known, the structure is unknown. We can easily develop the protein model to demonstrate the real world effect. So AI-powered target identification in computer, particularly the rational drug design approach. The main thing is you have to identify the target. Target means which enzyme or which receptor is responsible for the particular disease. So we have to find out the target. So that is your very important task in drug design. Once you find out the target, then we can decide the molecule which is having more affinity towards the target. That is the major part in drug design process. So there also we can use the AI. So that only in the beginning itself, I told the AI is utilized in all the steps of the drug design and discovery. So here the network analysis, it is maps protein-protein interaction. So that is first we should know what are the proteins which is involved in the interaction and which one is responsible for the disease. So for that, first you have to understand the protein-protein interaction in our body, how the two proteins are interacting. Then we have to find out the pattern recognition, identify the disease signature, means which protein is responsible for the disease, how it is modified or mutated, how it is responsible for the particular disease. Then target prediction. So by using this data, we have to find out the protein which is trackable. Trackable means we can easily design the track to target the protein, that is trackable protein. So you have to find out that protein. So AI system analyze biological network to predict disease associated targets. They integrate genomic, proteomics, and clinical data to identify promising intervention points for drug development. So this way the AI tools are used in target identification. So AI in virtual screening, just now I told virtual screening, it is a type of technique like what we are doing in high throughput screening. High throughput screening is a wet lab technique, means in the lab at a time by using the sophisticated instrument, at a time we can screen 100 or thousands number of compounds by using the bioassay. So there also, if you are using machine also, there is a cost of the chemicals, the time, and everything will play a role. So instead of that, and you have to have the synthetic library, 1,000 compounds, you want to do the HDS screening, you should have all the 100 compounds in physically. But it is not possible. Always you have to synthesize and keep the compounds for a long time. So what do you have to do? We have to do virtually. We have to design the molecules virtually. Means by using some software like drawing software, we can draw the structure, optimize the energy, and store it. So whenever the target is identified, so you have to develop the method, whether it is pharmacopoeial modeling method or QSAR method or docking method. By using that, we have to screen virtually what are the compounds are having some affinity towards the particular target. If it is showing, then we can take that molecule and do the optimization. Optimization means we change the functional groups according to the information what we are going to get from the computational method. By using that, we can improve the biological activity of that molecule. Meanwhile, reduce the toxicity and improve the biological activity, improve the ADME properties. All these things we can do. So that way, the virtual screening is most important in drug design process. There also we can use artificial intelligence. For example, traditional method, just now in computational aided drug design, that is docking method. It is speed is, it needs some time. But when you are using machine learned, so when we are doing docking, so you have to prepare the protein, you have to prepare the compounds, then you have to prepare the tracking parameter files, then you have to do the docking, then you have to do the interpretation. All these things you have to do one by one. But in machine learning, it is already trained one. So once you give the things, it will automatically do all these things, whatever I told. Automatically, the process will be carried out and we will get the result means 10 to 100 times faster than the docking method. So here the method is random forest method, what they are using in machine learning method. Then deep learning. There they are using three-dimensional convolutional neural network. Just now we have seen convolutional neural network. So this is 100 to 1,000 times faster than the docking method. Then hybrid AI recently they are using, that is multimodal method, that is 1,000 to 10,000 times better than the docking method. So now you understand why the artificial intelligence is most important and needed things in the current pharma field. So instead of doing one component by using simple computer-aided drug design method, we are implementing the artificial intelligence. Suppose we are doing in 30 days, we can do it in 1 hour time. That is the advantage. So AI dramatically accelerate compound screening. Deep learning models can evaluate millions of compounds in minutes instead of months. That is the advantage why we have to use artificial intelligence. So de novo drug design is nothing but from the scratch we have to design, because we don't have any idea about the drug. Just you are having the target, so you have to design the drug from the beginning. Means from the basic nucleus, then what are the basic nucleus you needed, what are the connectivity you needed, what are the functions, everything you have to design from the beginning. That is called as de novo drug design. There also we are using the artificial intelligence. So generative models create novel molecular structure from scratch, use variational auto encoders and transformers to generate diverse compounds. So by using artificial intelligence, the other method is adversarial and autoencoders. So here the generator and discriminator networks against each other. So they are having one generator and discriminator. Generator, it is using the old data, it is going to generate or create the new molecule. That is the function of generator. So when you are competing this generator and discriminator, generator is going to create the new molecule by using the old information. What the discriminator will do, it will distinguish this newly generated realistic compound with the old compound. So how far it is better than the old things. So that will do the discrimination, means it is going to distinguish these two. So combinedly both generator and discriminator network against each other. So it will give you a better and realistic product. So produce increasingly realistic drug-like molecule. So that is the advantage of when we are using AI in de novo drug design. Then here we are using reinforcement learning also. Optimize generated molecules for specific properties. Optimize novelty with synthesizability and bioactivity, because designing the molecule is not the matter. By using any techniques you can design, but the major important thing is whether it is feasible to synthesize or not. If you are design the molecule, it is showing 100% biological activity, there is no toxicity, it is 100% ADME properties, everything is acceptable, everything is okay, but it's not possible to synthesize, it's not feasible to synthesize. And what is the purpose of designing that molecule? So the synthesizability, that is the major important things when you are designing the molecule. So designing is not that much big matter, we can easily design, but the synthesizability is the major things what we want to concern. So the artificial intelligence will take care about the synthesizability. That only I told, when we are utilizing the artificial intelligence in drug design, it will optimize multiple number of parameters. It's not like human, we can go one by one. When we are doing conventional method, we have to go for one by one. Even we are using computational drug, computer-aided drug design also, we have to do one by one. But when you are using like machine learning, deep learning, reinforcement learning, the process is multiple optimization processes possible within short period of time. So AI-driven QSR model, already I told QSR model, when we are having number of compounds with its biological activity, then we can develop the model by using regression or multiple linear regression or partial least square regression. We can develop the model, then by using that model, we can predict the biological activity or the toxicity of the compound. Even we can predict the property relationship also. We can do in QSR. There also we can use AI, the artificial intelligence. So the accuracy, the prediction accuracy compared to the normal computer-aided drug design, the AI-driven is having better prediction accuracy. The R-square value exceeding 0.9 for many targets. Because whenever we are doing normal QSR modeling, the QSR, the R-square value, the correlation coefficient value, sometimes we get 0.9, some of the compound we can get 0.5, 0.6. But when we are using AI method, maximum times it will give more than 0.9. Then future extraction, deep learning automatically identifies relevant molecular descriptors from raw structures. That is the another added advantage. When we are doing computer-aided drug design, so we have to do all this regression method, simple regression method or multiple linear regression method. You have to change the model, you have to check whether it is valid, then we have to validate all. But when you are using the deep learning, it's automatically extract the information and find out which parameter is highly influencing the pharmacological activity or toxicity. So that is the added advantage. Then performance kinds. So this is the most important things. So you see the graph when we are using AI models and normal QSR models. So the reduce in prediction error by 30 to 50% compared to classical method. There are about 50% reduction in prediction errors. So when we are doing normal computer-aided drug design, there is possibility for errors. So in that 50% will be reduced when we are using the AI-driven QSR modeling. Then another important thing is protein structure prediction. Just now I told when we are using the AlphaFold method, that is DeepMind's AlphaFold, achieved breakthrough accuracy in Cas14. It predicts protein structure with near experimental precision from amino acid sequence alone. So just using the amino acid sequence, it will develop the protein structure. It is look like exactly the near experimental structure. So that is possible by using the DeepMind AlphaFold AI tool, and we are using. Then drug discovery impact. So enable structure-based drug design for previously undruggable targets. Means already the targets are known, not known. So that time we can use these techniques. Then we can develop the model and we can make it as a druggable target. Accelerate discovery for proteins with unknown structures, improve understanding for disease mechanism. So next one is AI in molecular dynamics simulation. This is the most important thing, because when we are doing simple molecular dynamics simulation by using the supercomputer or whatever it is, it takes sometimes a day or week or month according to the time scale what we are going to use. Suppose in nanosecond, 50 nanosecond after 20 nanosecond in a day. If it is 500, 2000 nanosecond, maybe 1 or 2 days. If it is in microsecond, then it will extend to, sometimes it is extend to month also. So that is the drawback in traditional molecular dynamics. So when we are using AI accelerated force field, the neural network potential reduce the computational cost by 100 to 1000 times while maintaining accuracy. That is the important thing. The accuracy wise, there is no compromise, but the time consumption, it will be reduced near about the cost, computational cost will be reduced near about 100 to 1000 times. So that is the added advantage when we are using AI in molecular dynamics. Then machine learning sampling, smart algorithm identify relevant conformational states, focusing computational resources efficiently. Then end-to-end AI simulation, deep learning directly predict protein ligand binding from structural input. Then AI assisted lead optimization. Just now we have seen the protein structure prediction. Now lead optimization. Why we have to do the lead optimization? That only I told earlier, when we are using any computational technology, whether it is a conventional method or high throughput screening or virtual screening or any other method, once the beginning stages we can find out the hit molecule, then we have to optimize the hit molecule to bring us a lead molecule. Means we can increase the potency of the molecule, that is called as a lead. Then the lead molecule we have to optimize again for the dose and the toxicity. All these things should be optimized, that is called as lead optimization. So we have to do the lead optimization. So when we want to do the lead optimization, we have to do some structural changes, structural modification on the surface of the molecule. So that will be done by any of the computer-aided drug design technology. But when we are using AI assisted, it will be faster. It will, already I told, it will optimize many things at a time. So optimize multiple parameters like potency, selectivity, and all together it will be optimized when you are using the AI technology, not like a CAD, computer-aided drug design. This multi-objective optimization was previously impossible with traditional method. Traditional method means both conventional method and computer-aided drug design method. It is not that much feasible, but here now it's feasible because of the use of artificial intelligence. Then another thing is natural language processing and discovery. This is another type of AI. So literature mining, AI extract relationship from millions of publication, identify hidden connection between drugs, target, and disease. See, now the best example is many of review articles are publishing nowadays. How? Yes, everyone is using the AI tools. Many of the people, more than 60 to 70 people, they are using the AI. So earlier, if you want to write a review article, you have to collect the articles, 100,000 articles. Then you have to select which articles to be included, which articles to be not considered. Then all the articles, suppose out of 1,000, you have selected 200 articles, all the 200 articles you have to sit and read and collect the information, and you have to synthesize it, then you have to rewrite it. So it will take 6 months to 1 year to write a review article. But nowadays it's not like that. Everything is, there is a AI tool. Just within a day also you can write a review article by using the AI. So that is the, I don't know whether it is good or bad, but the things are going up like that because the technology is day by day, the improvement in technology is unexpected improvements are there. I mean, unexpected things are coming because of this AI. So literature mining, we can do it. The knowledge graphs, we can do it. Then scientific insights. So it suggests novel hypothesis based on text analysis, accelerate hypothesis generation and testing. Everything it will do the software because it is trained like that. So AI in retrosynthesis planning, because sometimes retrosynthesis. So when we want to synthesize a big molecule, it's so size-wise it is very big. So when you want to synthesis, you have to go in backward synthesis. Means you have to fragment the molecules and make it into small molecules. Then you have to start the synthesis from that small molecules to the big molecule. That is the backward synthesis. That is called as the retrosynthesis. There also the scientists are using AI. So first you have to find out the target molecule. It is a complex drug candidate, so like rifampicin. It is a very big molecule. So how to synthesize it? For synthesis, it means already we are having the synthesis method, but the yield is less and synthesizing process is very tedious process. So we want to make it simpler. So we are using the retrosynthesis method. So by using the AI, we can find out the retrosynthesis process. So break down into simple precursors. So one by one, just break down. Then start from the very simpler particles, which is easily available and which is not toxic, which is environmental free. Like that, we can design, take the starting material, then move further. But minimization of step, we can reach the final compound. Then reaction condition. So we can optimize the reaction. We can predict and optimize the reaction condition easily with the help of AI. Then synthesizability assessment. That is the important things already I told. The synthesizability. So evaluates practical feasibility. Suppose you have done the fragmentation, you find out the simplest smaller molecules to synthesize the particular drug. But how we are going to synthesize? What is the process? That is whether it is feasible in the laboratory or not, or in the industry or not. That is the major part. So that is also evaluated by the artificial intelligence. So in all the way, the artificial intelligence will help to do the pharma things easily. So combining AI with high throughput screening, just now I told high throughput screening, it is your what you can tell wet lab technique. So when new target is identified, then you have to develop the bioassay. Once the bioassay is developed, we are having the library of synthesized compounds, thousands of synthesized compounds. Just we can use the sophisticated instrument, do the bioassay against the particular target and find out which chemical compound is having activity against the particular target by using the sophisticated instrument, that is high throughput screening. So there you can combine with AI. So then the technique will be more accurate and we can get the realistic value. So smart experiment design, that is AI select a diverse compound subset, maximize information gained from each experiment, adapt based on real-time result. Then efficient testing, that is another important thing when we are using AI. Reduced required experiment by 80 to 90%. Uses active learning to focus on informative compounds. Then it's extrapolate to understand the chemical space. So the time reduction is major, 80 to 90%. It reduces the recorded time. When we are doing in high throughput screening, high throughput screening is combined with AI, then the time requirement for the experiment reduction is 80 to 90%, means only 10% of the time we are going to use. Suppose 1 hour you are using for the high throughput screening, this will be done by within 5 to 10 minutes. Then AI in precision, we have talked only about the drug design, the medicinal chemistry part, how we are going to find out the target or design the target, how to find out the active packet, then how to design the molecule, how to synthesize, how to predict the ADME properties. That this all these things we can see the pharmacology, medicinal chemistry part and technology part. The same AI, it is used in the other side also, the hospital, pharmacy, clinical pharmacy, everywhere the AI is used now. Even now the dispensing also, now they started using the robot dispensing. On the operation in hospital also, now they are using the AI technology maintained robot for the operation. So AI is also used in the personalized medicine or precision medicine. So personalized or precision medicine means first you have to find out the genetic characteristic of individual. So according to the individual genetic characters, we are going to give the medication. Because suppose I'm having fever and another one of my friend also having fever. So both of us, 500 paracetamol is not going to work. For me, 500 paracetamol, 500 milligram, the paracetamol tablet 3 times a day is sufficient. But for him, he need more because his genetic makeup is different. So he needs 650 milligram 3 times a day. So that things we can decide by the biomarkers. First, we have to find out the biomarkers and the genetic information of each and every individual. But when we are going for individual genetic information, it's very difficult to manage by human beings. So we need the computation technology, particularly we need the artificial intelligence, because we want to do it very fast, very less time and more accurate. So we have to use AI. So that is they are using in patient stratification. That only I told. Suppose different people are having different genetic information. So when you want to make it as a group, so all the genetic information has to be analyzed. Then only we can go for stratify, means we have to split the groups. Next B, C, like that. You have to stratify the groups. So for that they are using AI techniques. Then response prediction. There also we are using the AI. Then biomarker discovery. That is the important things. The biomarkers is responsible for the particular disease. So there also we can use the AI techniques. Then AI powered drug repurposing. So this is the techniques what they have used during the pandemic, the recent pandemic COVID-19. They have used this technique, AI-powered drug repurposing. Repurposing means the drugs which is already approved for some other treatment by the FDA. Suppose a drug which is approved for AIDS, or the drugs approved for anti-cancer. That drug we can utilize it for some other disease. Because what is a drug? It is a chemical compound is having lot of functional groups. Because our body is also made up of lot of protein and receptor, everything is functional group. So there will be when we are taking a single drug, it's not going to interact with only one receptor. It is going to interact with multiple receptors, multiple enzymes, multiple proteins. So whenever you are having drug, so it will interact with multiple target, the poly target. So by using that one, they have come to the concept that is drug repurposing, means it is already approved for anti-cancer, but now we are protecting the other property. Instead of anti-cancer, it is also used for antiviral property. So the drug is also useful for to treat the viral infection. That is called as drug repurposing. So there also we are using the AI technology, artificial intelligence technology in drug repurposing. So what is the advantage when we are incorporating the AI-powered drug repurposing? Cost reduction. It is 10 to 15 times. There is a cost reduction compared to new drug development. So what is the problem in new drug development? We have to start from the scratch. But here already we are having the drug. It is already approved by the FDA. So no need to do the synthesis, the preclinical trials, all these things no need. But after some time, after some stage of the drug designing process, you have to start the clinical trials and other things. Because already we know the dose, what we know the safety. So all these things, first clinical trial 1 and 2, there is no need. 3 and 4 we have to do when it is coming for repurposing drugs. So the cost will be reduced, then time will be saved, because no need to do the synthesis and no need to go for the clinical trial 1 and 2. So 3 and 4 is sufficient. So we can minimize the time, we can minimize the cost also. Cost reduction will be 10 to 15%. Then the time, important thing is time. 50% time we can save compared to the new drug development. When you are going for repurposing, the 50% reduction in time. Then the safety profile, the another most important known safety profile, because it is already used as a drug, already approved by FDA. So it is, we know the safety. So 90% safety profile is there for the repurposing drug. So AI analyzes transcriptomic responses and protein interaction network to identify existing drug for new indication. So this is the techniques what they have used for the recent COVID-19 pandemic, and they came out with the drug within 3 months. After the disease was identified, within 3 to 6 months, they came out the drug for the treatment because of by using this drug repurposing technology. So this approach has already yielded several repurposed treatment for COVID-19. Otherwise, you think about that, the mortality. Within that 3 months itself, we lost many. But if they didn't find out the drug for the pandemic, then we cannot imagine what was the mortality. So explainable AI in drug discovery. So interpretability need. So whenever we are using the AI in drug discovery, the interpretation is most important. We have to understand what the AI is telling. But if we are lacking in that interpretation, then it's very difficult to do the things. So understanding of AI decision is most important. Then regulators demands transparency in drug development process. Then visualization method, attention map, highlight influential molecular features. So the future attribution techniques explain property prediction. Then trust building, another important things, explainable models gain scientist acceptance. Clear reasoning supports regulatory submission, because the regulatory is another important things. We have to get the approval for the drugs. But when we are using the AI in drug discovery, we have to get the approval. So we have to satisfy the regulatory requirements. If it is not satisfied, then we cannot bring the molecule into the market. So AI prediction, predicting drug-drug interaction, the another important things when we are taking multiple drugs. When we are under the medication of multiple drugs, there is a lot of possibility for drug-drug interaction. So experimentally, when we want to optimize all these things, it is again a time-consuming process. So by using the AI models, we can predict the potential interaction between the two drugs by analyzing molecular structure and metabolic pathways. That is the another important application of AI in drug design process. Then AI-assisted formulation. So we have seen all these things. Why not in formulation? Yes, the AI also assisted in formulation development, particularly in pre-formulation studies, and what is the dose. Want to decide, we can use artificial intelligence, and which formulation we want. That also will be decided by AI, and it will give idea. So formulation prediction, AI predict optimal excipients and ratios, consider drug properties and delivery requirements, means which delivery route we want, whether it is parenteral, oral, whatever the things. So we can, using the AI, by using the molecular structure of the compound, it will decide according to the experience and other things, it will decide which route is better for the particular drug. Then stability optimization model focused long-term product stability, identify formulation with extended shelf life. The shelf life is, how much it is important for the pharma products. So that also we can extend the shelf life. Then bioavailability enhancement. So another important things for better potential, the bioavailability of the drug, means the availability of the drug in your biological system. So the AI will suggest strategies to improve drug absorption, then optimize dissolution profile. There also we can use AI. Then not only this, in clinical trial also we are using AI. So the patient selection, how we are going to select the patient, particular patient. So according to the information, the biomarkers obtained from the patient, the artificial intelligence will analyze and it will give the suggestion what are the patient will selected for this clinical trials. Then adaptive design, the designing method, the algorithm adjust trial parameters in real time. Then outcome prediction, that also will be done by the models produced by AI. It will forecast trial success probability. Then real world evidence, it will compare with the real world evidence. So quantum computing and AI drug discovery. So the quantum advantage, the quantum computers solve molecular simulation exponentially faster than classical system. That is the advantage of the quantum computers. So it's enable the precise calculation of quantum properties of drug target interaction. So we will get better result than the normal one. Then we can use the hybrid approach, means we can combine the quantum computing with AI in drug discovery. Then it will boost up the drug design process and we can go for high level optimization. Then future prospect of this integration promises unprecedented accuracy and speed. So we can do the things fast and the accuracy will be better. So early application focus on quantum chemistry and protein folding. So then AI-driven antibody design. So we can use the AI in antibody design also. So it is useful for drug prediction of the structure prediction of antibody. Then affinity optimization, we can do it. Then immunogenicity reduction, we can analyze it. Then developability assessment, whether it is feasible to develop or not. So all these things we can optimize by using AI. So AI in fragment-based drug discovery. So the fragment-based discovery means we can use the small molecule for the discovery, the fragments. So we can find out the fragments which is responsible for the particular target. So that also will be done by the AI screening. Virtual libraries to find the fragments that bind target packets. Means which small fragment is, means the small molecule fragment means small molecule, which fragment of the particular the small molecule is having high affinity towards the active packet. The target packet means the active packet of the target, either it is a protein or receptor. So that one we can do with AI. Then binding mode prediction, we can find out what is the binding mode, how it is going to bind with the protein. So we can find out the binding site. Then we can optimize the fragment to improve the binding site, because it is having somewhat binding site, but we want to improve the binding affinity. So for that, we can do the fragment growing. Fragment growing means we can optimize the fragment to make it more binding towards the, increase the affinity towards the particular active site. Then fragment linking. Suppose we are having two fragments, both are showing the activity against the particular target. Then we can link these two fragments and make a single molecule. Because it is fragment means small molecule. So we can join these two molecules. How to join? What is the procedure? So all these things the AI will suggest. So AI for predicting protein-protein interaction. That is another important things. Why we have to find out the protein-protein interaction? Because in the body, one of the protein is going to bind with the another protein. Then only the biological process will start. So which one protein is, where is the problem, where is the issue, which is causing the disease? We have to find out whether the protein A or B is responsible for the particular disease. You have to find out. So for that, you have to do the predict the protein-protein interaction. So from that, we can easily come to know which protein the target is responsible for the disease. Then we have to design the molecule for that protein disease causing interaction, that is here. So not only this, the AI is also used in toxicity prediction. That is the most important things, because whatever the drugs, we cannot tell any one drug which is free from toxicity, because the reason is already I told, whatever the drug you can take, everything is a chemical which is made up of a lot of functional group. The functional group is not going to interact with only one receptor. It is going to interact with multiple receptor. It is going to interact with multiple proteins. So whenever it is going to interact with the protein or receptor which is causing the disease, then it is the expected activity. We can get the expected activity. When it is going to interact with the other than that, it is going to interact with the other protein or receptor, definitely it is going to produce the unwanted effect, the side effect, or it is also going to produce the toxicity. So that also we can do the prediction there. Nowadays, there are number of tools, online tools are there to predict ATMET properties, absorption, distribution, metabolism, excretion, and toxicity. There are number of tools. All it is running behind this AI technique only, either it is machine learning or deep learning techniques. So AI powered chemical synthesis. So already we have seen it is used in the retrosynthesis, that is the backward synthesis. Now we are seeing the AI powered chemical synthesis. So reaction prediction, automated synthesis, novel reaction, all the way the AI technique is used. So the federated learning in drug discovery means where we have to combine the learning, join the learning techniques in drug discovery, then it will be better output. So local model training, parameter sharing, global model aggregation, and model development. So all these things we can share except the data which is private. Suppose you are using the data to develop the model, you can share the model but not the data, because it is a private data. We cannot share that data, but you can share the model that will be utilized by your partner, those who are your joining partner. They can utilize it, they can improve it, they can utilize for some other prediction. So you can, the sharing is most important. Whatever the research, not only here, but particularly here, the getting the data is very, very difficult. So whenever we are using the data, it is a private data, we cannot share, because we are going to use the biomarkers, the genetic biomarkers. We cannot share it with others. So that is private. So that should be keep with the, those who are the main owner of that one, they can keep. But once you develop the model, that model will be shared. So that model will be utilized by other partners in the team. So that is important. The federated learning in drug discovery is most important when you are talking about AI. Then AI in pharmacovigilance. So there also we are using AI. So to find out the adverse event detection and signal validation and regulatory reporting. Everywhere nowadays the AI system is there. Earlier you have to write the report and submit, but now everything is online. Just you have to feed in. Everything is processed and it made is by the AI. So challenges in AI driven. Yes, I have told everywhere from the beginning to end up to pharmacovigilance, we are using AI. But what are the challenges we have to face when we are going to use the AI? Model interpretability. That only I told. The AI is going to give the model, but we should know how to do the proper and correct interpretation. When we are not able to do the correct interpretation, then everything will go wrong. So understanding AI decision remains difficult. Sometimes it will give the models which is not explainable, which is not understandable. So first thing is you have to understand the AI decision. Then data limitation. That only just now I told the data, we cannot share some of the data, we cannot share some patient data, then genetic information, we cannot share with others. So that is the other challenge in when we are going for AI. Because when we are having more number of data, then we will get better models. Suppose we are having 1,000 data, instead of 1,000, I'm having 100,000 data. That will be better. The model will be trained properly. So the training is most important. Those who worked in QSR model, they know how the model, when you are developing the model, how to train the model. And now you are having more number of component training set, then we will get better model with better validation properties. So that is the important thing. Then regulatory uncertainty. We have guidelines for AI designed trucks. We are having lack of all these guidelines. So the regulatory bodies, they have to come with the guidelines for AI designed trucks. That is the need for now. Yes, everywhere the AI is entering all the field. So they have to develop the guideline for AI designed trucks. Then technical integration, combining AI with existing workflows. Just now we have seen how AI with the Dino, how AI with the clinical trial, how AI in target predicting, how AI in track means the chemical compound optimization. So the combining, that is another important things. Then despite remarkable progress, significant challenges remain in AI-driven drug discovery. Addressing these issues require collaboration between, that is the important thing, AI expert. Yes, we are a pharmacist, we are not the expert in AI. So we need the AI expert to do the things. Then drug developers, we as a pharmacist and the regulatory peoples. Yes, as a pharmacist, we are not familiar with all the regulatory properties. Those who study the regulatory properties, they only know the regulation. So ethical consideration, this is the common things. So bias in training, data, transparency requirement, data privacy concern, accountability question. So all these things will be considered in a drug-driven discovery. So the future direction, so we can go for a multimodal system, autonomous lab, digital twins, and quantum enhanced AI. That is the combining just now I told. So that is the hybrid technique. So we can implement all these things in future direction to make it faster and to give the realistic data, more accurate and precise data. So you have to go for all this direction. So these are some case studies I have given. What is the successful of the AI? So in AtomNet by Atomwise, that is used deep convolutional neural network to identify the potential treatment for Ebola virus. So this is the one where it is used to find out the threat for Ebola virus. Then AlphaFold, already we have seen. Then in silico medicine, that is GAN-TRL, which was developed for the fibrosis treatment in 21 days by using AI application. Then Apcellera's AI platform, which is used to find out the candidate for COVID-19 in just 3 months. So these are the some successful AI application in computer-aided drug design. Then another drug, which is Halicin, that is AI discovered antibiotic with novel mechanism active against drug-resistant bacteria. Then the another medicine, which is target idiopathic pulmonary fibrosis, that is INS 018055. Then AlphaFold application, which is used for enabling untrackable target. So the best practices, so start with clear objective, build multidisciplinary teams. That's already I told who are there, who will be there in the multidisciplinary team. Then invest in Tetra infrastructure, create validation and framework. So these are the some important practices when we are implementing AI in drug discovery. The same thing. So the economic impact of AI in drug discovery. So the traditional cost when you are developing a drug, you have to spend $2.6 billion. So the cost reduction is 30 to 50%. The time saved is 1 to 2 years less than that. Then return of investment, it is 10 times better than the traditional method. Then future of AI drug discovery. So AI is in transferring every aspect of drug discovery. Just now we have seen what are the different stages in drug discovery. We are using AI in all the stages. Integration with experimental methods, create powerful hybrid approaches. Then there is a paradigm shift moving from sequential to parallel drug discovery, equality, truly personalized medicine development. So there we can use it. Then call for action, invest AI literacy for drug discovery teams, develop regulatory framework for AI design drugs. That is the need of now. Because just now I told in all the process we are going to use the AI. So there is a regulatory guidelines we need. So foster open collaboration with academia and industry. That is the another important things. So thank you for all for your listening for the last 1 hour with your patience. Dr. Mukesh.
Just a cabinet position. Thank you very much, sir. Yes, now we have interviewed me and we were. Yes. Yes.
Thank you, sir. This was a very informative session. We trust that the audience has gained valuable knowledge. Sir, please accept this e-certificate as a token of our appreciation.
Ladies and gentlemen, we call the proceedings as of now for a tea break. Delegates and participants are requested to move towards C block foyer area. All are requested to assemble in seminar hall C block again at 3:50 p.m. sharp for the valedictory ceremony.
Thank you.
Was at 3:50.
Huge round of applause. Gentlemen, you may please be seated.
The best is yet to come, celebrating our achievement. A very warm good evening, Honorable Deputy Director, Professor Dr. Mukesh Singh Sikarwar, Organizing Secretary, Neeraj Mishra, Head of Department, faculty members, staff members, our valued delegates, and dear students. I, Abid,
And I, Roshni, feel immense pride to stand here before all of you this evening on behalf of this Amity Institute of Pharmacy, Amity University, Madhya Pradesh. We are delighted to offer you all a gracious welcome to the valedictory ceremony of the National Conference on Artificial Intelligence in Drug Design and Drug Delivery and Design, from innovation to implementation.
now I would like to invite Professor Dr. Neeraj Mishra, Head Department of Pharmaceutics, Amity Institute of Pharmacy, to summarize the conference. Sir, please.
Good afternoon, all of you. Dear students, on behalf of Amity Pharmacy, Amity University of Madhya Pradesh, I, Dr. Dinesh Mishra, once again welcome you all the students and participants who join the online and offline mode in the conference on the artificial intelligence in drug delivery and design, from innovation to the implementation. So today, the first guest speaker, chief guest of today's conference, Dr. Manish Kumar Chaurasia, who is a senior principal scientist, CDRA Lucknow. He will give the idea regarding the role of AI in the identification of target molecule. He will also discuss about the role of how the AI, important role of AI in the reducing the cost of the clinical trials, and also help in the identify the target molecule, and also help in the lead molecule. And Dr. Chaurasia also discussed regarding the how the AI can be used for the scale up and post-approval changes that SUPAC, and to identify the best formulation, so that can be ease of work of the formulator. So in the future era, he told that the AI can be used if the use can be very judiciously, and definitely going to reduce the cost of the drug designing, because it will reduce the extra burden of the CAD. So we can easily get the target molecule, and that will reduce the cost also. The keynote speaker of the today's function, Dr. Sayur Jain, who is the head department of pharmaceutics, Naipa Mohali. He's also discussed the advanced elaborate role of the artificial intelligence in the drug delivery. Nowadays, we are saying that the AI is used in the robotics. You see, the China is very advancedly developed and designed the model of the AI there, and AI is used in case of the for the searching purpose also. And then now the self-driven car also there by the AI, and robotics also. That is, we can say it is a upcoming feature is the AI. But my question is how we can control the AI for the mankind. So we should use he also discussed about the role of AI in the nanotechnology, and also discussed regarding the role of AI in the quality drug design and the clinical studies, clinical trials. So ultimately, you can say, if the scientists working in the various fields, if they use the AI in the precisely in the proper manner, definitely we can cut short the cost of the drug in the target molecule. And third speaker, that is keynote address given by the Dr. V. Ravichandran, who is the head of chemistry, MS University, Malaysia. He joined the online mode. So he has discussed the actual role of the AI in the CAD. He discussed how the AI can be used for the designing the target molecules, and how the AI can be used in case of proteomics to design to identify the proper structure of the target molecule. So in future, we can say that will easily can design the target molecule, and we can easily do the drug targeting, and we can easily locate the drug site. Ultimately, we can do the targeting in the very precise manner. Now come to the conference remarks that we have received in the conference about around 450 registration, who is on the out of which around the 295 join online and 160 join offline, out of which 130 from Amity and remaining from the outside. So we have received the total 105 abstract from various part of the institutes across India, and out of which we have received the 36 oral presentation, including online, offline, and 6 poster, and 6 which can be 6 also offline and 6 online. So total we have received around the 50 presentations, including the oral and the poster presentation. So at the end, I would like to thank all the members of all the committee, including the scientific committee also. So it is thanks for myself. Thank you.
I respectfully request our Honorable Deputy Director, Professor Dr. Mukesh Singh Sikarwar, to present the appreciation seed to Professor Dr. Neeraj Mishra, the Organizing Secretary, in recognition of his exemplary contributions. Sir, please.
I would like to invite all the professors to kindly grace the stage for the honor of distribution of prize.
Now the time came to felicitate the judges of oral and poster presentations. For this, I request organizing secretary along with the professors to kindly felicitate Dr. Bhagat Singh Jainwal. Now I request Dr. Rajendra Chauhan to please kindly come up on the stage for his felicitations. Now I request Dr. Alok Soni to please come up on the stage and receive his felicitations. Thank you, sir. Now I request Mr. Abhishek Pathak to please come and receive his felicitations. Now I request Mr. Ankur Agarwal, please receive his felicitations.
Talent and skills are the crop that are nurtured, groomed, and given all possible opportunity to grow and grow. With this quote, let us proceed towards the most awaited event of the week. It's time to honor the exceptional performances of the participants of poster presentation and oral presentation that we have witnessed today.
Let us commence with the announcements of the winners of the poster presentations. The third position goes to Ms. Muskan Gupta. Please come up on the stage and receive your prize. The second position is backed by two participants, Mr. Somdat and Mr. Satish Kumar. Most awaited first position, that is the winner of the poster presentations competition, is backed by two participants, which is Mr. Ajay Jadhav and Mr. Harshit Verma. The winners of the online poster presentations are as follows. The third position is backed by Gopi Reddy Tejasvi. The second position is backed by R. Sai Yasvita. And the first position is backed by Mayank Sharma.
Mentioning the winners for oral presentation in online mode. The third position is secured by Vikas Singh. The second position is a tie between Bensi Baby T and Patel Todika Sohail. And the first position is secured by Purnima Agarwal. Now let us move towards the declaration of winners of oral presentation in offline mode. The third position is secured by Ms. Shivani Yadav, Amity Institute of Pharmacy. Please have a huge round of applause. For the second position, it's a tie between two. Ms. Shivangi Sharma and Ms. Shivani Prajapati from Amity Institute of Pharmacy. Put your hands together for the first position holder, who is Mr. Arun Pachori, Parul Institute of Pharmacy, Parul University, Vadodara.
Cherished faces of the winners is something worth capturing to boost their enthusiasm for further events. We kindly request all the winners to assemble on the stage for a group photograph. Please join all the winners on stage.
All the posters and oral
All the posters and oral presentation winners are requested to please come on stage to have a group photograph.
Now, we would like to invite the Deputy Director, Amity Institute of Pharmacy, Professor Dr. Mukesh Singh Sikarwar, to propose a vote of thanks. Sir, please.
Hey, good afternoon, everyone. Good afternoon, all the dear participants, and online or offline. Dear, everyone, please. Good afternoon, everyone. It is my privilege to give the vote of thanks on behalf of MD Institute of Pharmacy. And I'm very glad to offer the vote of thanks because this event has been planned, cherished, and executed with a lot of thought process, a lot of hard work by many members, and of course, the success depends on many people. So I'm here to acknowledge the hard work of all the people who are behind the success of this conference. So first of all, I would like to give the profound thanks to our Honorable Pro-Chancellor Sir and Vice Chancellor Sir for giving their consent to grace the occasion. So they were there in the morning in the session, and they were continuously present throughout the session. So my sincere thanks to both of them. My sincere thanks to even Registrar Sir, he was there throughout the conference. And my heartfelt thanks to our chief guest, Dr. Manish K. Chaurasia Sir, who is the Senior Principal Scientist, CDRI, for giving their consent and gracefully giving the talk in the, in our conference. And his kind presence was very benign to our conference. So thanks to him. And many thanks to the speaker, Professor Dr. Sanyog Jain Sir. He is the head of the Department of Pharmaceutics in Naipur, Mohali. It's quite far. He traveled all the way from Mohali to join us here. Thanks to him. And my heartfelt thanks to Professor Dr. V. Ravichandran, who joined us from Malaysia. He's the head of Pharmaceutical Chemistry Unit, AIMS University, Malaysia. So, sir, presentation was very exhaustive and very informative. So if sir is there online, thank you, sir, for your kind consent to attend the conference and give your consent to be the speaker, to be the keynote speaker in our second session. Thank you, sir. Sincere thanks to Professor Dr. Manoj Mishra, sir, our HOI, for the kind guidance and the encouragement and support for executing this conference. And I'll be failing my duties if I do not thank the organizing secretary who is sitting over here, Professor Dr. Dhiren Mishra sir, who has worked extra mile, sleepless nights to make sure that every aspect of this conference goes well and we carry out the conference and everything is taken care. And my sincere thanks to all the principals, HOD, professors and faculty members from various colleges in and around from Gwalior, and even I'm glad to see the participants has come all the way from Baroda, from Chhatarpur, from many. How many are there from Baroda and Chhatarpur and other places? Please raise your hands. So we were very glad to receive the participants from outside. So thanks to you, to the faculty members. Thanks to the sponsors. We do have a sponsor. Our sponsor was UV Scientific Hyderabad for their support. They kindly gave the sponsorship to our conference, and we have duly acknowledged him. And then, thanks to all the presenters who gave the presentation, oral and poster presentations. Thanks to all the delegates who have joined the conference in person and online, because we had so many delegates who joined online and so many delegates who joined in person. My sincere thanks to all the participants, and thanks to all the judges, juries for the oral and poster presentations. And we have so many other departments to support us throughout this conference. We had ESCO department, I think still there. Some of them, they are taking photographs. For media, taking photographs, for capturing our events. Thank you very much for taking the photos and video coverages. Thanks to IT and AV team from Noida, as well as from our AUMP campus for Zoom link, and for providing the IT support for make sure that the event goes well online as well as offline on the stage. So thanks to IT and AV team. Thanks to all the committee members for the excellent support, because this was not possible without them. And I have a long list of the committee member. Almost all our faculties were there in all the committees. So I'll just name few committees please accept my apologies, because time is running out and we're short of time, so I cannot read all the names. But thanks to all the committee members who are here present, committee, especially scientific committee, we had registration and finance committee, then we had technical and IT committee, hospitality and transportation committee, then discipline, reception and certificate distribution committee, then media and publicity committee, then we had stage and mementos committee, then we had sponsorship committee, and we had purchasing and printing committee. So all these committee members, heads are here. I think the members are here. My heartfelt thanks. Thanks from my bottom of my heart, because it was not possible without the hard work of all these committees and the members. Now, thanks to all the non-teaching staff, supportive staff was there, even cleaning, non-teaching staff was supporting all the time. So thanks to them, and thanks to all the moderators. Wonderful moderation done by all the moderators, anchors in the, both the stages. We have 2 stages in the auditorium as well as here. So very well taken care. Thank you. And thanks to caterer for the food, tea, excellent lunch. Thanks to in charge for both the blocks, Block C and Block B, for providing the support for various events which we park in both the. Even thanks to the team who made the Rangoli. I think if the students are here, please raise your hands if you are there. We can clap for you. There was a Rangoli made by some students. Thanks to them. And thanks to all the students for participation. Without you, this conference was not possible. Even though the students were there who didn't do the registration, but they wanted to learn. They wanted to see what is going on in the AI, and they were very much patient, listening to us, listening to all the, the speakers from outside. They were there throughout the day in the auditorium. They were here, and still many of them, despite the time is over, they may even go, because they were allowed to go after 3 o'clock normally, but still they are here. So we can give a big hand to all the students who are here. Thank you very much. And thanks to the security AUMP for making sure that everything goes well. Thanks to all the students for participation, and thanks to all the people who directly and indirectly supported this conference. So at last, thanks everyone. Stay safe and happy journey for those who are going, and enjoy your day. Thank you.
A successful event like this always calls for a group photograph. For this, we kindly request all the dignitaries, faculty members to assemble on the stage for a group photograph.
So this is the final day. Thanks. Alright, I have already captured. All done.
Volunteers also, volunteers also.
Okay.
After that, sir, after.
Right. I think even we can make the last announcement of the sponsorship, because it is obligatory for us. Please, you can make the announcement.
The end of the story is the new beginning for many others. On behalf of Amity Institute of Pharmacy, we sincerely thank the delegates, dignitaries, faculties, staff members, student volunteers, and attendees for being with us this evening and enlightening us with their insightful remarks. Special thanks to our sponsors, UV Scientific Hyderabad, for their valuable sponsorship. We also shower our heartfelt gratitude to all the attendees at this conference.
The student volunteers after this photograph are also requested to be present here for a group photograph.
Okay, we are taking photos now. What we can do is we can take the photos in batch wise. Like once this photo is over, some of you can come and take photo and then move. Then some of you come batch wise and we can take all the photos for everyone. Thank you.
We request the student volunteers from all the committees to please come and have a group photograph. The student volunteers from every committee.